So I'm going to briefly introduce you. So welcome, Bastian. Yes, thank you, Benno. So Bastian Reick is a principal investigator of the IDOS lab at the Institute of Artificial Intelligence for Health and the Helmholtz Pioneer Campus. And he focuses there on machine learning methods in biomedicine. And before joining Helmholtz, he was at the Machine Learning and Computational Biology Lab at ETH Zurich, and he obtained his PhD in computer science from Heidelberg University, where he also got his maths degree. So maths can bring you very far, right? So Bastian, the floor is yours, and we're looking forward to your talk. Sorry for the delay.
Oh, no, absolutely not. Thank you very much for the kind introduction, and thanks for having me. The title of my talk is a little bit whimsical, but I hope that we can have a nice discussion about this. So I'm going to talk about good gradients and how to find them towards multi-scale representation learning. And I guess that this talk is a little bit different maybe from what you might be used to, because I'm talking about a relatively new field that is just beginning to be developed and to be researched upon in machine learning, and that is this whole idea of using multi-scale representations or of using underlying fundamental structures of a data set. And this is actually my forte in some sense, because I used to be trained as a topologist, and in topology, in mathematics, we focus on these underlying structures of data quite a lot. And I'm going to give you an overview into the field and zoom into to two applications, one from colleagues of mine that have been doing this for a while now, and that have been looking at the topology of viral evolution. And the other one is a little bit more recent and is from my own research together with students and my postdoctoral advisor, Professor Borgward from ETH. And in this, we are presenting a new architecture that I think will be useful for driving and advancing this field. Now, onwards. By the way, one administrative thing, I actually can't see the gallery of people. So if some of you have a question, I would kindly ask the moderator to just interrupt me, because I just can see the slides at the moment and nothing else.
If it's okay, we will be collecting the questions at the end of your talk. So
oh yeah.
Okay. Yeah.
Sure, of course. So already at the very beginning, just to do some terminology. So when you hear manifold and when I hear manifold, we might be thinking about different things. So this talk is not about this specific manifold in that sense, but this talk is about some more complex or some more mathematical shapes, such as this Klein bottle on the left-hand side, which you might have heard about, or something very weird that is called the Alexander Horn sphere. So all of these objects that are flying around in mathematics are what we call manifolds. And the term itself, I would say, is seeing a renaissance in quite a lot of fields, but often it's not really discussed, or it's not really intuitively defined what people think about. Some people think that any data is kind of a manifold, so data is becoming equated with manifold. Some people have a more distinct definition there and only think about different data sets. So let's see how we can align all of those. Now, before we go really into this, let me give you a brief background in topology. Before we zoom into manifolds, which are a little bit more complicated, let's first talk about topology. In topology, so in mathematics, the study of abstract shapes of objects is known as algebraic topology, if you have a more algebraic perspective, so counting things, or differential topology, if you're interested in a more functional perspective and you want to evaluate things on those spaces. Now, these objects can look very complicated, as I showed you this funny sphere before, but let's just take a look at some very simple, almost exemplary objects that we often find. This would be a cube here, a sphere, and a donut or a torus. I should stress that this is not one of the donuts that you would like to eat, because the torus, in this sense, you can't see that, of course, but when when we talk about a torus in mathematics, we think about having this thing not filled with anything. So basically it's just the outer surface, and it's like a cylinder that you can fill with air, for instance, and that you close then up. So it's not a filled donut, unfortunately, because I'm mentioning this for technical reasons, because if some of you have been familiar with this a little bit for any reason, then you would find that the torus, if it were filled, would behave a little bit different than from what I'm showing here. Anyway, now let's take a look at the first cube. The cube looks a little bit weird in the sense that it has sharp edges, whereas the sphere doesn't. But from the point of topology, those two objects are kind of the same, because when you think about the cube, you can sort of start removing some of its edges and start sanding it down in a sense, and then end up transforming it into a sphere. So from the point of topology, the sphere and the cube, even though they look a little bit different to us, they are not fundamentally different. They are, of course, they look a little bit different, but fundamentally they are the same object. It's just a little bit, one is a little bit more smoother than the other one. However, the torus, on the other hand, is clearly a little bit different from those two other objects. Primarily, and this cannot be stressed enough, primarily because it has this huge hole in the middle. So it features a certain characteristic that the other two objects don't have. And so it should be treated a little bit different than the other two objects. And this is what the whole field is about. The whole field is about taking a manifold or manifold-like space and seeing what makes it different from other manifold-like spaces. Now, to give you an informal definition of what we consider when we talk about manifolds is, we would say any object or a space that locally looks like some d-dimensional Euclidean space is a manifold. Now, that means that when we talk about the Euclidean space, you just have to think about the usual, well, coordinate system that you know from high school or from drawing plots or stuff like this. So you have a two-dimensional coordinate system, for instance, or a three-dimensional coordinate system with three different axes, x, y, and z. And so having a d-dimensional Euclidean space just means that you have d-independent coordinates to describe the position. And now when we say locally looks like some d-dimensional Euclidean space, this is to be understood as saying if we really zoom into the object, then we would expect that it's sufficient to have d dimensions in order to describe the position. So let's see an example of this. If we just take a circle, and notice that I have fattened up the circle a little bit to make it a little bit more visualizable here. If we zoom really, really into that circle, then we basically just have an infinitely thin line, of course, and we can't really move away from this circle. So we only have a single coordinate that describes our position. It can be seen as kind of maybe the angle, or if we arbitrarily say that this is 0, for instance, and we move in the positive direction here, and this can also be seen as the distance to this arbitrary origin, with the understanding that at some point it might just wrap around. But in any case, we just have a single independent coordinate to describe our position. Now, going one step higher, with a nice sphere, we have an increase in dimensionality, meaning that if we now zoom into this sphere, we would anticipate that we need two independent coordinates to describe our position, because even though there's a lot of people around that believe that the Earth is flat, I mean, maybe not a lot in total numbers, but still more than you would anticipate given our scientific advances. This is kind of true in the limited sense that when you really zoom into the Earth, it really looks flat. You don't see the curvature of the Earth if you're standing on it, because it's just so vast. And this gives you an inclination of why we only need two independent coordinates to describe our position on this sphere. Or alternatively, if you would like a more physical description, you could also say that it's sufficient to have two coordinates, in this case, one angle and a second angle, in order to describe the position on the sphere. So you can see that these intuitive and this more coordinate-based definition, that they line up. Now, For the torus, we have a similar thing. We only also need 2 independent coordinates to describe the position, because as long as we don't zoom out too far from this space, we will also just see a locally planar 2-dimensional object when we're standing around it. So, of course, there's more manifolds around. So basically, whenever you have a very smooth function, for instance, you can bet that this function somehow generates a manifold structure or is somehow related to what we would call a manifold. The only limiting assumption I would have to make here is that let's just ignore the boundaries for now, because when you add boundaries to your space, then it becomes a little bit weird, and handling these boundaries becomes a little bit more complicated, because you have to account for what happens when you approach them and when your object certainly ends. We don't have this issue with the sphere so much, because we can always walk around the sphere, but for this, for instance, we would kind of fall off this object when we approach this boundary here. So this makes it a little bit more problematic. So for all intents and purposes, let's just ignore those boundaries and just think of all the statements that I'm making in this talk to apply to the interior of that space. So the bottom line, or the takeaway from this brief digression into topology, should be that manifolds come in relatively many shapes, forms, and sizes, but they are quite a natural description for many of the phenomena that are happening around us, because when we assume that we are given some samples from a data set and going into nature and collecting some experiments, it's a very nice idea to have an illustrative model, a kind of Platonic ideal of the space that we are dealing with in mind. And manifolds can constitute such a model. And again, let's just try to bridge this gap a little bit here conceptually. When we're dealing with real-world data sets, we cannot expect to find the manifold, because real-world data is inherently noisy when you collect it, and so there's all kinds of biases going on. The measuring equipment might be faulty or might be probabilistic in a certain sense. You might only find entangled measurements. You might not be able to account for confounding factors, for instance. All sorts of things plague the data analysis in the real world, of course, but conceptually we might often want to think about having this very Platonic idea of a manifold as our kind of model space. And this is illustrated here when we sample from a torus multiple times. This image will come later again in a different context. Now, one thing that is really becoming increasingly relevant in data analysis, and that you might have stumbled upon now, is this so-called manifold hypothesis. And this is actually just a very fancy way of saying that many people hope that a real-world data set can often be adequately described by one or multiple manifolds, meaning that we assume that we have the sampling procedure, and if we were to do a lot of experiments and account for all the confounding factors and so on and so forth, then we would be able potentially to describe a very nice mathematically idealized space, like this manifold that I showed you before, from our data set. And oftentimes this hypothesis is really not that far-fetched. So I mean, otherwise this whole field wouldn't work, of course, but there are a lot of examples where you can really see the intuition behind that and see why it makes sense for a certain object to be described like this. So for instance, when you take pictures of a natural person and you vary the light source position, and the light source is allowed to like rotate around a fixed axis, for instance, then these sort of pictures could ideally be construed to live on a very low dimensional manifold, because you're changing only one single parameter in the image. So despite being potentially very high dimensional, you would assume that there is a very nice underlying structure of a low dimensional manifold. And that gives us hope to do real world data analysis tasks, because oftentimes in the real world, our data sets are extremely high dimensional and the things we want to describe are extremely low dimensional, or rather our visual apparatus for dealing with the descriptions in the data sets is incapable of visualizing or handling more than 2 or 3 dimensions anyway. And two questions are directly translatable from algebraic topology to modern data analysis, and that is how to distinguish between manifolds. So if I do multiple experiments in the wild, do I always get the same manifold? Do I get samples from the same manifold, or do I get something different? And the second would be how to classify manifolds. So can I say something like, okay, I would expect that there is a certain class of manifold that appears more often than another class. We'll now see how to tackle those in principle using multi-scale approaches. Now, all of this is borrowed from a very simple taxonomy almost in algebraic topology, and that is known as the so-called Betti numbers, or Betti numbers if you want to pronounce it a little bit different. It's intuitively always called that you essentially count d-dimensional holes in your data. So for instance, if you have this circle here, you would say that, okay, it has a zero-dimensional hole. A zero-dimensional hole would be a connected component, and it has a one-dimensional hole because you have this whole circular structure here. So you would assign this data set the Betti number signature of 1, 1. Now, if you go a little bit higher, you would say in the sphere, you also have a single connected component. This time you have no circles whatsoever because there is just a surface, but this surface encloses a void, so you have a Betti number of 1 in dimension 2. So this one has a signature, a Betti number signature of 1, 0, 1. Now, going a little bit higher, with the torus, you also enclose a void, so you have a Betti number of 1 in dimension 2. We're going to skip that dimension for a second, and we also have a single connected component because the torus is obviously a connected object. You can move from any point in the torus to any other point, so that one is also pretty much given. But now let's go a little bit deeper and see what happens in the second dimension, in dimension 1 actually. Why do we have a 2 here? Well, it turns out that there are two different circles or cycles of this data set that you can describe topologically. One is the obvious cycle, I would say, namely this one here. So you can take the torus and put it on your finger like you would wear a ring, essentially. That is one of the characteristic cycles. The other characteristic cycle is a little bit harder to see, but you can easily imagine it when you think about taking this torus, and I mean, it's almost Christmas time, right? Taking this torus and putting it on a Christmas tree. If you put it on a Christmas tree, you would take some string and you would wrap it around the torus like this. This is working. Hopefully you can see the cursor else. This doesn't really make a lot of sense, but you can wrap it around like a piece of string to hold it and to put it into your Christmas tree. And this is the second characteristic cycle of this data set. And since all the cycles, so all the positions into which you could put this piece of string are kind of equivalent to each other, so there's no well-defined origin of the torus or no well-defined holding point of the torus, we don't count these infinite cycles more than once. So basically we just say, okay, there's one cycle that is characteristic like this, and there's another one that is characteristic like this, leading to a signature of 2 in this dimension. Now this might sound a little bit weird, but if you want to revisit this later on, let's just think about that the Betti numbers are a very simplistic way of counting connectivity in a data set. So we don't have to fully understand these numbers in order to make use of them. Because what we can do with them now is we can use them as simple signatures. So instead of taking a look at the sphere or at the torus and saying, well, they look a little bit different, we can say, well, we have this mathematical descriptor, and the mathematical descriptor yields different outputs. So by necessity, the datasets must also be different. And this is the whole idea, or the whole spiel in algebraic topology, I would say. Finding good invariants, finding good descriptors of datasets, and then pitting them against each other and seeing what you can say about your data in practice. Now, there's quite some ground to be covered before you end up doing practical inferences though, but we'll get there. Now, in reality, of course, we're not dealing with these idealized shapes, but we're dealing with a data set. So we just have these points here and we need to think about what we can do with those. And I should stress that this is a really ingenious construction that you're about to see here. It's not due to me, unfortunately. It has been around for quite a while now. Essentially, it goes back to almost more than 100 years, I would say, where this was described for the first time, but it only recently became leveraged in mathematics, especially in applied mathematics, and people understood that, well, this is actually something that we can do now with computers, and we could actually use that to describe our data sets. And the ingenious idea is to say, if you're given some points here, you can best describe the shape by just approximating the shape at different resolutions, so at different kinds of zoom levels, if you will. And these zoom levels have a natural analog in mathematics. The zoom level is obtained by growing Euclidean spheres around every data point and then connecting data points whenever those two spheres overlap. That's kind of the natural equivalent to a scale, and I'm hammering this point in because it's an equivalent because it mimics what happens when you squint at the data set or when you look at it from different scales. So you're essentially giving an individual data point more weight and blowing it up, pretending that you have sampled things in between. And then as soon as these spheres start touching each other, you would connect them by an edge, or if 3 of them touch, you would connect them by a triangle. If 4 of them touch, you would connect them by a small clique in a graph, and so on and so forth. And you can repeat this process, as you can see, you can repeat this for multiple resolutions for an increasing resolution number, and you obtain much more fine-grained information about the space. In particular, you can see that for a certain resolution, namely this one here, for instance, you can actually see a circular structure appearing. And this circular structure is still present in here, but it's gone when you zoom out too far, when you have basically turned your data set into a full blob. So this last figure on the slide, you have to think about seeing this data set from very, very far away. So the higher the epsilon parameter, the further away you are from the data. And when you are very close, you see the individual points up close, of course. But when you are very far away, you only see a single blob. So these data points start merging into each other. And this is what this ingenious process is supposed to simulate. Now that's all well and good. And I can understand that maybe you're not sharing this mathematical enthusiasm. But the nice thing is that you can now calculate these individual Betti numbers for each of the resolutions of the data set and obtain kind of a multi-scale view of how your connectivity changes. So let's do this, and let's run through this a little bit. So you start with epsilon 0, then you increase it a little bit, nothing happens. You increase it a little bit further, you get your first connections, because remember, the circles are touching now, so you have to connect them. You go a little bit further, you still have the circular structure in here. And you go a little bit further, and now it's gone again because you zoomed out and it's a single blob. So this can be visualized essentially by following this line of the Betti number here along this simple plot. So for low resolutions, you don't have a circle, you don't have a circle, then you have a circle. This circle remains for quite some time, and in the end, it's gone again. So this also shows you that if you were to pick a single resolution with which to evaluate this this data set, it would be hard to do so because it's a little bit unclear what a good resolution would be. And so again, the ingenious mathematical idea was to say, well, let's pick all the resolutions and just do a multiscale descriptor. So instead of saying, we have we do a cut here and we have a circle, or we do a cut here and we don't have a circle, why not create a descriptor that incorporates this idea that at certain resolutions you have one circle and at certain other resolutions you don't have a circle. And this is the whole idea of persistent homology. And I know that homology is unfortunately also an overloaded term, and it's used in biology, so I would say in the more serious sciences, and not just for mathematicians who want to have some fun. But in topology, homology refers to a generic operation, a generic way of associating what we call a sequence of algebraic objects, such as abelian groups, to other objects, such as topological spaces. So this sounds a little bit weird, but the idea is just you take a sequence of something and you map it to a sequence of something else. And you do this, of course, in order to make the object that you're looking at a little bit more understandable. Now, in the previous example that I showed you, you would end up collecting these multi-scale numbers, these Betti numbers that are changing over the course of filtering the data set. You would store them in what is called a persistence diagram. So this is a multi-scale topological descriptor. And the idea is that points that are stored in there represent a topological feature, for instance, a circle in the data set, and their distance from the diagonal in this data shows you how persistent they are. So whether they endure or live for a long time in this filtration process, in this filtering process, or whether they are destroyed early on and are just kind of short-lived. Now, as a very simple example, the persistence diagram from this previous data set, which basically, if you remember, it was just a few points arranged in a roughly circular shape, it consists of a single point. This is consistent with our idea because there's only a single circle in there, which appears at certain resolutions. It appears at resolution 0.2, and it vanishes at resolution 1.0. So this is why this circle gets assigned a coordinate of 0.2 and 1, meaning that we have captured its appearance and disappearance in the data set. And you can see how the data looks at these resolutions by mapping it back into this diagram. So at 0.2, we see the circle for the first time, and at 1, it's already finished. Now you have to believe me that for smaller values than 1, there's still kind of a circle around. This is of course because I chose the I chose the numbers to make sense here. But this should give you a small idea, a small inkling of an idea of how this is supposed to look. We will also see some examples of more complicated data sets in a few minutes. Namely, this also works for functions over the real numbers. Now, why would you do this for functions? Well, when you're analyzing functions, it's often nice to have an idealized model where you say, oh, this is what my signal is supposed to look like. And I can associate that with this persistence diagram, with this interesting topological descriptor on the right-hand side. But in reality, I never measure this nice smooth signal, but I measure something different. I measure a very perturbed signal here that might have smaller oscillations, smaller oscillatory behavior. And I can do the same process and assign this persistence diagram also. And you can see that, interestingly, the large-scale features are preserved in this diagram. So they look astonishingly similar, except for all this, well, junk, I would say. It's the junk DNA of topology, if you permit me the quip here. Except for this junk at the diagonal, these two diagrams are more or less identical. And this points to a deeper understanding or to a deeper intuition behind these methods, namely that they are able to incorporate noise into the calculation, and instead of ignoring the noise, which some methods try to do, or dealing with the noise by filtering it out or by doing something else with it, it just incorporates the noise and just lets you as the end user decide whether to incorporate the noise or not. So in this case, we would be hard pressed to say, oh well, these are obviously proper features because we see that they correspond to these oscillations here, but in real world, in other applications, this might be different. Now you can also do more complex examples. So this whole idea of similar processes give rise to similar diagrams. This also holds in higher dimensions. So when you sample, for instance, multiple point clouds from a torus in three-dimensional space, you can see that despite the point clouds being relatively different in terms of their shapes, because sometimes you just don't end up sampling with the same density here, you can still see that the overall shape of the persistence diagrams is roughly similar. And this again points towards a deeper understanding of looking at the data set from a more fundamental perspective. And when I say similar, I mean this looks very qualitative, right? This is we obviously want this to be more quantifiable in order to do some calculations later on. But this can also be arranged, of course. So there's a whole bunch of methods dealing with calculating distances between those topological descriptors. And all these distances are based on the theory of optimal transport, where the idea is that you take one probability distribution or general distribution of things and you map it to another distribution of things while accounting for differences in size. Now, it's optimal transport. I'm not going into the details here, but there are well-defined optimal transport measures between the persistence diagrams. You can think of them as creating a perfect matching between the two diagrams, with a caveat that you have to account for the noise somehow, for the points that you don't want to match. So this optimal matching algorithm must have the option to send points back and to say, well, I can't match those and I incur a cost for it. But the bottom line, the takeaway message should be that these descriptors, these persistence diagrams, can be calculated. They are stable to perturbations in the data set, and they are robust to those perturbations in the sense that if you have a continuous perturbation, a continuous update of your data set, your underlying descriptor also changes continuously. And the last point, you can actually calculate distances between those descriptors. Now, let me show you a brief application here that I found fascinating. This is not this is relatively old work, I would say, from 2013. I mean, in mathematical terms, it's still super fresh, but in machine learning terms, it's from the dark ages. But it's a very interesting paper. The idea that the paper is called Topology of Viral Evolution, and please don't ask too hard questions about the actual evolutionary aspects in there, because I'm not a biologist and I can only appreciate the biology from afar. But what I get from this paper is that it's about assessing the evolutionary behavior of viruses. And the paper is particularly interested in what they call a reticulate evolution. So if I understand this correctly, this is horizontal gene transfer, and I really hope, since this is recorded, that I'm not saying anything super wrong here. What they are doing is they're performing an analysis of these of genomic sequences. And now they're looking at the topology of those genomic sequences in order to see whether horizontal evolution is different from ordinary evolution. And they actually find that horizontal evolution results in non-trivial 1D topological features. So 1D topological features would be circles or cycles. And so they're capable of finding these cycles in genomic sequencing data, which I find fascinating. Again, I can't walk you through all of this because I'm not entirely familiar with the background of this work, but I wanted to reproduce one of the plots here, which I found fascinating. So those are the two main plots of the paper. One for what they call the vertical evolution. These are genomic sequences that have been projected into, these are distances between genomic sequences that have been projected into two-dimensional space using PCA. And this is the corresponding persistence diagram. And now you can see that the horizontal evolution point cloud has a radically different appearance, of course, but also a radically different persistence diagram, a radically different topological descriptor than the vertical evolution sequences. And this is fascinating because when you think of it like this, these diagrams on the right-hand side can always be calculated. They don't have to be calculated from two-dimensional projections. So you can actually calculate them directly from the distances between the genomic sequences without having to use a different embedding algorithm. Now, this is exciting for me because if you pick an embedding algorithm for your data, you also incur what we call an inductive bias. This means that you pick an algorithm, and by picking the algorithm already, you are prescribing certain properties to your data set, and it might not be clear whether those data sets, whether those properties actually hold in practice. And so, yeah, this is why I'm pretty excited about this technique, but it's just a sneak peek of what is possible. And we'll now rather do the deep dive into deep learning. Namely, it's all well and good to calculate this from some smaller point clouds and learn something about the underlying phenomena. But what we really want to do is we want to put this into a model and have a model that is capable of telling us whether certain topological features, certain topological structures in a data set are actually relevant for us and whether they can be used to obtain better embeddings, for instance. And I'm going to give you a brief, very, very brief, short overview of autoencoders here. So this is just to have some shared terminology. I'm not sure how familiar you are with autoencoders already, so hope this is not too uninteresting. But an autoencoder is a general neural network architecture in which you put in some input data that is d-dimensional with a big D. You put it into an architecture that compresses this data into a d-dimensional representation with small d, which is called the latent representation. And then you blow it back up again to end up at big D many dimensions again. And this is the reconstruction. Now, this might look a little bit weird at first glance, but it actually makes perfect sense because typically this input data set dimensionality is much, much higher than the latent representation dimensionality. And so what you're doing is you're forcing your data into like a compressed version of it, and you try to reconstruct the data later on from this compressed version and assess to what extent you can still reconstruct it and how qualitatively different these reconstructions are. And these qualitative differences are measured using a loss function, and this gives you an overall quality of the reconstruction. Now, why would you want to do this? Well, the autoencoder is a classical, what we call encoder-decoder architecture. So you encode it first into the latent space, and then from the latent space, you decode it again. So what we're doing essentially is we're learning the identity function of the data set. So we're just learning a map from the data set to the data set itself, but with the added caveat that we can use this middle layer as a bottleneck, as a compression technique, or as a latent representation. And these latent representations have all kinds of interesting properties. We can use them for visualization for once, of course. We can use them for interpolation. So if we map our data points into a different space that we understand a little bit better, we can, for instance, say, suppose I have two data points here and I connect them with a line. What data points would actually correspond to the middle point, for instance? So we can generate some new data points in this latent space and reconstruct them back to an original image, for instance. We can also do clustering, of course, and many, many more applications. So I really want to stress that this is a very generic, very high utility neural network architecture. And there's a whole bunch of architectures around there, so you can do this in all kinds of shapes and forms depending on your application. A classical, or well, I should say, a well and often used architecture, for instance, is the so-called variational autoencoder, in which you learn the parameters of a distribution that tells you how to actually generate your input data or how to generate new samples from the input data set. So there's a lot of powerful things here, and I urge you to look into that paradigm in case you're interested. Now, the simplest autoencoder that you can build, and I actually also have code for this, so if someone is interested in learning more about those, I can also hit you with this repository. It's actually very, very easy to do nowadays, thanks to very good libraries. The simplest autoencoder is a linear transformation from d-dimensional space into two-dimensional space. The decoder is a linear transformation back, and the loss function is just the mean squared error. So you take your reconstruction x tilde and you check how far away it is with respect to the squared Euclidean distance. And I'm using this as a point to not to say that this whole field is very, very simple, of course, and there are much more complicated architectures, as said before, with a variational autoencoder, but at the same time, this should also give you an understanding of how to think about these sort of components of the autoencoder architecture in an intuitive manner. So the loss really just measures how well your reconstruction matches the original data, and encoder and decoder can be anything you want, but for instance, linear transformations are pretty good, or convolutional transformations could also be employed. And now towards the end of the talk, let's move to one of the applications, one of the exciting things that I was able to do in my time at ETH. So this is our paper Topological Autoencoders, together with Max and Michael and my postdoctoral advisor, Carsten Borgward. And here we wanted to tackle a very, very interesting and relevant topic, I would say, namely, when you do dimensionality reduction, so when you take your data set and you compress it into a low dimensional space, you need some characteristic information about the underlying manifold structure in order to produce faithful embeddings. So in order to produce embeddings that don't just, well, show you things that aren't there in the original data set. And this is important because we have a very simple running example where we were able to demonstrate this. Suppose you are taking a very high dimensional data set that consists of a very big sphere. And in this sphere, you have a nested set of smaller spheres. So it's kind of like these Russian nesting dolls. You have one big sphere, and then in there you put some other spheres that are not intersecting. And now you try to visualize this with a normal standard autoencoder architecture. And what you get is you get something like this. So notice that the colors are coming from the indices, from the identities of the spheres here, because of course we know what we put in, and so we know which points to colorize in which color. But what you can see here, and what the takeaway message should be here, is that the standard reconstruction, the standard latent space in your standard autoencoder, is completely ignoring this nesting structure. So it just takes the big point cloud, the big sphere, puts it somewhere. It puts some of the smaller spheres almost inside there. So if you, without the colors, you wouldn't be able to see that there are these clusters around, and it botches up the scale differences. So for instance, it makes this sphere here and this sphere here look much smaller than they are in reality. So you're essentially getting a representation that is not appropriate for inferring structural information about the data set. I'm not saying that this latent representation is necessarily wrong, because it's possible that you can perfectly reconstruct the data set from this. The only thing that you can't really do is, if you rely on this latent representation to be used in other contexts, then you are in a very, very problematic domain. And to tackle this, we added a secondary loss term to this aforementioned loss term of the autoencoders. So our main idea is that we want to preserve topological features in the latent space. So we want to ensure that the latent space has a similar topology than the original space. And we're doing this here schematically by taking the input data set, calculating its topological features, so ending up with these persistence diagrams that I showed you before. We're also calculating the persistence diagrams of the latent space, of the latent code representation. And since the persistence diagrams are always comparable because they live in the same dimension, we can compare those two spaces by means of the persistence diagrams and obtain a topological loss term. And this is completely independent from the reconstruction loss term that we have in a standard autoencoder architecture. So we can weigh those two together and obtain an overall loss that weighs geometry and topology to a certain extent in order to obtain a good reconstruction. And now, in the interest of time, I'm probably not going to talk too long about the actual loss term implementation, because that requires some additional technical details here. But suffice it to say that the loss term that we developed in this paper is actually bidirectional. So it measures kind of the loss that you have to endure if you go from the input space to the latent space, so from X to Z, and the loss that you have to endure when you go from the latent space to the input space back again, so from Z to X. So it's a dual component, it's a bidirectional loss term, and that is actually what makes it work for us. So we initially experimented with a unidirectional loss term where we were only looking from X to Z, and this one didn't work so well. The nice thing also for you, the takeaway message should be from this slide, that these loss terms are not that hard to implement in practice. I mean, we have code for this already, but they're really just based on very simple Euclidean distances between topological structures. So there's no optimal transport needed here. I want to stress this emphatically because it would otherwise make the method very, very slow. Now, let me show you, towards the end, let me show you some practical results and how this looks qualitatively. I'm sparing you the quantitative evaluation. For this, I would happily refer you to the paper itself. We were able to use a whole bunch of different quality metrics in order to assess to what extent topological and geometrical features have been retained, and the topologically regularized autoencoder, or short TopoAE, our method consistently scores very, very high there, so it has some good properties. It also doesn't negatively affect the reconstruction loss too much, even though it has a slight effect on the reconstruction quality, because now, of course, our goal of maintaining certain latent structures is not necessarily aligned with having good reconstructions. So those are two different goals. I need to stress that this is a little bit to be expected. But in a qualitative evaluation, where we just are throwing the same data set with the large sphere and the smaller spheres in between, where we're throwing this at the data, we find that our topologically regularized autoencoder is capable of maintaining the size differences and maintaining the nesting differences. So it puts all the spheres at more or less the same scale. It shows that there is a larger sphere surrounding them, and it doesn't rip anything apart in the latent space. And you can see that this is quite distinct from the behavior of other architectures or other algorithms in other contexts. So for instance, UMAP kind of distributes the larger sphere to the individual spheres here, but it cannot maintain the spherical shape, at least not with the parameters that we found using cross-validation. TSNE is similar. It is capable of maintaining the spherical shape a little bit better, but it also kind of rips apart the bigger sphere. And again, the topologically regularized autoencoder is the only one capable of maintaining these separations in the latent space. Now you can also see these differences here when we zoom in a little bit, so you can see that indeed the spheres in the topological autoencoder framework are maintaining their relative sizes and are placed in the correct spots, if you can call it that. And the nice thing is that going from left to right, you only had to add our topology-based loss term to your autoencoder architecture. So basically, this loss term is fully compatible with whatever autoencoder architecture you can come up with. You only have to make sure that it is properly weighted during the optimization procedure, so that the original loss term and the new topology-based loss term can be given either roughly equal weights or some different weights depending on your application. So, and that's it from my side. I want to outline a certain thrust for the future that I would find very interesting, or that I would love to see implement in the community. There's actually two strands that I would like to work on, or that I would like to point to you. So there's a whole strand of topology-based algorithms in which machine learning algorithms are augmented using this. So these are novel algorithms that incorporate some topological concepts in machine learning, and the autoencoder, for instance, is a very, very nice idea of this, but there's also a bunch of other ideas around to make this work a little bit better. But what this really should go towards, and this is what I'm now working on at the AIH, and this we're hopefully ramping up the research group soon. I want to use this to drive personalized medicine applications. So I want to imbue personalized medicine with multi-scale ideas from topology. So for instance, having access to multi-scale representations of a complex data set in order to solve a prediction task for patients, that would be one of the goals. And I think that the topological autoencoders framework and other frameworks can be extremely used here. And last, I want to stress that topology is really, really good at unlocking these heterogeneous data sets. So when your data is heterogeneous and you don't have a single defined scale, then this is something where you can use topology with very, very good results. If you have a single defined scale, there's not that much use in using a multiscale perspective, of course. So let's reserve this for the cases where you actually have multiple scales and you want to capture more than one at the time. So one of the strands that I'm working on at the moment with this framework is the extension of the topological autoencoders to single cell datasets, because those are very, very tough to analyze still. I mean, you know this better than I do, and they're also highly heterogeneous, I would say, because they because you have different scales, you have different noise levels, and you have different measurements and sometimes even different modalities that need to be matched. So that's it from my side. Thank you very much for your attention, and I'm happy to take questions now.
Thank you very much. This was really exciting, bursting with questions, but I'm sure there are some questions from the audience in Paris. If you have a question, yeah, I think so. There are a few people here who are working with individual specific networks where you have a network representing a single individual. So where the edge weights are really individual specific. And there are some people in the group who are trying to, okay, also embed this, you know, in an embedding space, but with classical autoencoders. So I think here, because we are losing so much of a topology and we are really interested in the topology there, that might be an added value as well. Yeah. What about time course data? Do you see that also as a different scale?
Yes, it's a little bit more complicated then, but one thing that I would also really like to break into, I just haven't mentioned this here because it's a whole new topic. Would really like to break into time varying data here as well, because then you immediately have a different axis there, right? So you could either look at the scale at a temporal scale or at a spatial scale, or at a mixed scale of both of them, right? So something that happens in spatiotemporal with spatiotemporal differences. So that's really exciting to me. Yes, absolutely.
Yeah. Okay. Yes, I do not see the audience myself. Are there any questions from the audience? I do not see anything on the chat, but we have mentioned to our participants, please keep your questions, and they can always send it to the organization, and we will transfer it to you, or maybe they know your email address and can send it directly to you as well.
Yeah, that would be happy. Yeah.
Okay. So if I do not get any message that there is another question, I move on to the next speaker. I see that Celia Greenwood has already arrived. So it's my pleasure to introduce Professor Celia Greenwood. She's a full professor at McGill University and had her PhD degree in biostatistics from University of Toronto. She's also co-director of the Ludmer Center for Neuroinformatics and Mental Health at McGill. And she's also been the president of the International Genetic Epidemiology Society, IGES, which is also one of the societies very close to my heart. Her research domain is in statistical methodology development for analysis of genetic and genomic data. And I think your lecture will also be in that same spirit. Celia, the floor is yours.
Thank you very much for the welcome. I'm going to see if I can share my screen. Okay, good. Well, thank you so much for the invitation. And I guess it's the afternoon, so I won't say good morning, although it still feels a little bit early here. It's a real pleasure to be with you today. And I wish I had been able to sort of be there in person and listen to all the other presentations, that's for sure. So here's an outline, tentative outline, depending on how time goes. I'm going to do some general concepts of genetic architecture, a little bit of GWAS, polygenic risk scores, and then some examples basically coming from research of some of my students. And then sort of go back again to architecture at the end in different populations, which is not something I'm going to talk a whole lot about. Christelle, can you tell me whether all the students have run across GWAS already in this week, in this workshop?
I would say that most of them know what GWAS is, but we are a very heterogeneous student population, more from the experimental to the translational side over methods. Yeah.
Okay. And I would welcome questions during if there's some clarifications that people want to ask about as I go along. Please don't hesitate to jump in. Okay. So like I said, I'm going to start with a little bit of sort of thoughts about genetic architecture, or a little bit of background on genetic architecture just to get going. And so we can think of that as the characteristics of genetic variation responsible for heritable phenotypic variability. So this is going to include the number of genetic variants that affect a trait, their frequencies in a population, the magnitude of effects, and whether they interact with each other and with environment. So I'm sure that these are concepts you've run across before. You know, we often divide the genetic architecture of a trait into these four sort of simplistic, oversimplistic categories: the monogenic, oligogenic, polygenic, omnigenic. The monogenic you would have run into in high school. I'm sure you have a dominant disease, a recessive disease, possibly multi-allelic, etc. And I can pick one example. One of the well-known ones would be phenylketonuria, where you're unable to digest phenylaniline. And if you're in this kind of a situation and you are trying to figure out what to do with that from the point of view of medicine, the medical system, you want to identify who's carrying it. In more generality, for any monogenic disorder, you want to maybe understand what's gone wrong, and of course treat. If I go to an oligogenic example, you can debate these for sure, which box some of these examples should be in. I've put serum vitamin D levels there because there are a small number of loci that have quite a lot of influence on your serum vitamin D levels that you have. There are other loci that have smaller effects, but there's sort of four that have quite large effects. I've also put cystic fibrosis in here, which you might debate because, of course, it's a single gene disorder, but there are modifier genes that affect the severity of cystic fibrosis. And so we can consider perhaps that we want to think of it as oligogenic rather than monogenic. Again, on the right-hand side, I would argue that with these systems where there's not so many loci that are contributing most of what's going on, we probably want to identify those loci, understand what they do, and figure out how to treat people who are ill or at risk of becoming ill. But then when you go into the polygenic category, of course, there's lots of examples, coronary artery disease or coronary heart disease, diabetes, many, many more. What you want to do with the information that you have about the polygenic information may start to change a little bit. Certainly, we still want to identify what are the risk factors. We may still want to understand what they all do, but if there are hundreds of them, it's going to start to become harder to figure out what all of the genetic contributors are actually doing, and how they're actually changing the cellular biology. So in this kind of a situation, you might start to change exactly what you're going to do with the information, and you might start to think about stratifying the entire population, or stratifying people that are coming into a hospital or into a clinic, and then trying to have different interventions based on the polygenic risk score. You might think about screening, for example. And so there will be treatment involved, but the treatment may not be targeted to any one genetic variant because there are hundreds going on here. So the questions you're going to be asking are going to start to change. And then, of course, if we go to the idea of an omnigenic architecture where pretty much everything plays a role, then understanding the contribution of one particular genetic variant or one particular gene is going to start to become nearly impossible. On the other hand, the secondary concepts of being able to stratify individuals or screen individuals, those may still be very relevant. So the argument that, the architecture will determine what you do is kind of where I'm trying to go today in this lecture. So that was the sort of the introduction, and now I'm going to go into genome-wide association studies, and then polygenic risk scores, and then examples. So I really have not very many slides on genome-wide association studies, and I'm glad to know that most of you have probably seen them before somewhere. But I do have a slide or two just to set the stage. So very basically and very generally, we're going to test association between a disease or a trait or a phenotype and all the single nucleotide polymorphisms or SNPs across the genome, one at a time. In assuming that there are 3 possible genotypes for any one variant, any one SNP, which I could, write as AA, AB, BB in a generic format. What is usually done is to code that in an additive model. So you score that genotype as a 0, 1, 2 by counting the number of copies of whichever allele is more rare in your population of interest. So now you have a numeric variable, and then you test for association with your phenotype, and then you plot the results and look at which ones are most significant. The most commonly seen plot is probably the Manhattan plot. And here I have just put one example of a Manhattan plot, and I'm sure that you've all probably seen many of these. This is a Manhattan plot for bone mineral density measured in the lumbar spine with the DEXA measurement. It comes from a 2009 paper. And I've picked this one of the many because I'm going to come back and talk about bone mineral density later in some of the examples. And because I actually think this is one of the ones that still looks quite like Manhattan. It has a few skyscrapers and then a background, whereas some of the plots that we see today where people are using huge cohorts like the UK Biobank, the skyscrapers are almost no longer visible because there's so many of them. But anyway, this is a plot where it still looks a little bit like Manhattan, perhaps. And so, of course, these p-values are graphed on the -log10 scale so that the small ones pop out at the top. So there, of course, have been thousands of papers on GWAS studies, and there are lots of subtleties involved. I'm just going to talk about a few of them. It's not the focus of what I really want to talk about today, so I'm not going to spend a lot of time. If you have a continuous phenotype Y, linear regression would be most commonly used. And here, oh, I don't know if you guys can see my mouse. Can you see my mouse? Whoops. I don't think you can see my mouse. Okay, that's fine. Yeah. What about if I do this? No.
Yes, we can. Yeah.
Okay,
I can see the arrow. Yeah.
Thank you. So I'm going to use G for genotype. That may come back in other slides. And beta is the effect of that SNP or that genotype or that per allele effect on Y. And, the genotypes could be simply coded 0, 1, 2, or they could be centered and scaled by the variance of the minor allele frequency. Another thing you may see in some of my slides, introducing a little bit more notation, is the idea of X being some other covariate that you might be interested in. Age or sex, for example, would be commonly used covariates that might go in. And so they would be added possibly to adjust for confounding or to capture additional error, to reduce the size of the residual error to give you a bit more power. So Y might need to be transformed so that your residuals are well behaved. These are standard concepts in statistical analysis. and some of the detailed parts that I won't talk about, is how do you decide which allele is the minor allele or the major allele, things like that. Those are things that are very important, but that I'm not talking about. If you have a binary disease, then often people will use a logistic regression. The notation is otherwise the same, but instead of having y on the left, you have the probability of being diseased. And again, x are additional covariates, and now the exponent of beta hat will be estimating the odds ratio for disease risk. SNPs that are not actually genotyped can be very effectively imputed by leveraging existing studies on sequencing, such as the 1000 Genomes Project, or the UK 10K study, or the HapMap Research Reference Consortium. In fact, it's really easy to do this. If you have a genotyping on a set of individuals, you can actually send those genotypes to one of these imputation consortia and get back many more millions of genetic variance estimates than you had originally genotyped. So again, I'm not going to talk more about imputation, but it's very commonly used, and it's really quite straightforward these days. One of the most important caveats with genome-wide association studies is the differences between ancestral populations in terms of the genetic architecture and sort of the consequent false conclusions that can arise. If you have a poor reference database for your imputation, you may be introducing biased estimates of the genotypes. If you have different trait prevalences and symptoms between populations, that can also confuse your results. So false positive associations can arise due to confounding quite commonly. Most genome-wide association studies will include a few principal components of genetic ancestry among the extra covariates to try and minimize those false positive associations. However, that's a partial solution to a very, very challenging problem, because if the architecture is very different across ancestral populations, and you analyze a sort of a combined set of individuals coming from different ancestries, you may miss important associations. So we have both false positive, the possibility of false positive associations, possibly arising from confounding, and we have the possibility of false negative associations by sort of hiding things if you have quite a group of individuals from a number of different backgrounds. So this is a really important caveat to genome-wide association studies. I'm going to come back really at the end, assuming timing is going smoothly, and talk a little bit more about that. But I should definitely point out that this is a very active area of concern and of research, and I think there's a lot more to be done there. So I had showed you earlier a Manhattan plot for bone mineral density from a 2009 paper. Here I'm showing you some of the results of a genome-wide association study for osteoporosis from 2017. So this was a study that was using one of the early releases of the UK Biobank. And what's plotted here, this is obviously not a Manhattan plot, what's plotted here is the minor allele frequency of the variance on the x-axis, and then the estimated effect, or beta hat, using the notation from my slides, on the y-axis. And the loci that were identified are colored in red if they had been previously found in an earlier genome-wide association study, and in blue if they were found in this GWAS and appeared to be novel in this GWAS. And if the locus had been identified as being close to a particular gene or linked to a particular gene, it's been given a name. The other thing you can see on this graph is a dashed line in black. The dashed line is essentially corresponding to where we anticipated to have good statistical power to detect an association. Statistical power is going to depend on the actual value of the beta, the true association between the SNP and the trait, and the minor allele frequency. And so that's why that line is curved. What you can very much see clearly here is that the newly identified loci in blue are lying very close to the dashed line. So basically the loci that are being discovered here are exactly being identified at about the power that this study had to identify those loci. And many of the loci that had been previously discovered, the red dots, are above the line, as in they had larger effects and therefore could have been discovered quite easily in studies that were smaller. The earlier studies in general were smaller. So I find this slide, really quite informative. It really shows clearly power, statistical power. It also shows how many loci there are, for this particular trait, osteoporosis, and this is in 2017. There are more loci now. It shows the huge range of allele frequencies that are involved. It shows that many of the loci have very small effects. So a lot of those estimates are around 0.05, so they're really very small effects. And you can see that the architecture of this trait as time passes is becoming more complex. The earlier 2009 Manhattan plot just had a few skyscrapers popping out the top. Here we have lots of loci going on. So let me go back to architecture. This figure is taken from a paper that I was involved with in 2018, talking about genetic architecture. Just to think about architecture a little bit in the abstract, these are examples, from real studies. We have on the left-hand side, we have type 1 versus type 2 diabetes. Loci that were known at the time of this publication are shown here with dots, graphing again the minor allele frequency on the x-axis and the odds ratio, or the sort of increased risk of disease, on the y-axis. Now, if you compare here the blue dots to the red dots, you can see that for type 1 diabetes, you have many, many more loci with larger odds ratios and with quite small minor allele frequencies than you have for type 2 diabetes. So for type 2 diabetes, we have most of the loci seem to have really quite small odds ratios down near the bottom. And there are no red dots over here on the left. So even though these two diseases are obviously related to each other in terms of their impact on metabolism in the body, the genetic architecture of the two traits is really quite different. And to take another example, on the right-hand side, vitamin D versus LDL cholesterol. So these are things that you can easily measure in the serum, that are quite commonly measured in the clinic. But you can see here that for vitamin D, there's really only 4 dots, which I mentioned earlier. There are 4 loci that really contribute most to the genetic architecture of serum levels of vitamin D. And in contrast, there are many, many dots for LDL cholesterol. So we have a much more oligogenic architecture for serum levels of vitamin D than we do for LDL cholesterol. So looking at these plots of the minor allele frequency versus the contributions or the beta hat estimates can give you a bit of an idea of the differences of the architecture of different traits. This is another picture from the same paper in the UK 10K study, which was published, I believe, in 2015. The loci that were identified for many complex traits had smaller effects, smaller values of beta than had been previously identified. So it was a larger study than many previous studies, and therefore had more statistical power and was able to identify effects that had smaller values of beta. And you can see in the two lines on the graph, the red line or the yellow line, that again, many of the effects that are being found in that study were very, very close to the line of statistical power. That's what those lines again are, statistical power curves. And so what you're going to find in your study is going to be strongly associated with the power of your study. So that's what I'm going to say about genome-wide association studies. I have a series of points going on through my presentation here. And so this is point number 1. GWAS have identified and are identifying many loci with influence on phenotypes and diseases. I didn't talk about fine mapping at all today. Fine mapping remains challenging. Of course, there's a lot of work on that. How do you go from a genome-wide association signal to understanding exactly what that signal does and which gene it's related to? That is a whole field that I wouldn't be able to talk about today in addition to what I have planned to talk about. But this is now a good point to ask if there is a question or two before I move on to polygenic risk scores.
I do not see questions on the chat.
Okay.
Mm-hmm.
All right. So, next topic, polygenic risk scores. So I showed this plot already, and I was talking about, how many loci there are, how many of them are of small effect. And so I think this plot is really illustrative of the question that has motivated the polygenic risk scores. So what can you do with that information? Now imagine that you have a highly polygenic trait with hundreds or thousands of associations whose effects are often very small, or perhaps the alleles themselves are very rare in the population. What can you do with that information? You can go and try and figure out what each locus does, which gene it's associated with. And like I said, that fine mapping step is very challenging. Even if you did that successfully, what are you going to do with that? Now that you have several hundred associations and you know which genes they're linked to, how are you going to take that back, into a clinical context? So the polygenic risk score is saying, well, I don't know how I'm going to use each of those bits of information separately, but perhaps I can put all that information together and try and think about prediction. So I'm going to try and predict who is at risk of disease, and I'm going to try and predict perhaps also who is at risk of, a bad outcome of their disease, or who is going to deteriorate quicker than somebody else. So I could say that as, okay, I'm going to want to build a model to predict my phenotype Y using my SNP data and any other information and covariates that I might have. What model should I use? It's one question that we can talk about. And I also have a sort of statistical problem that I have many more SNPs than I have people, particularly people with disease, in most studies, which is sometimes referred to as the P much greater than N problem in statistics, where P would be the number of SNPs or the number of covariates, and N would be sample size or number of people. However, that's what a polygenic risk score essentially is. I'm building a model relating Y to all the SNPs. So I can write this as I want to model Y as some function F of all my SNPs G. So what's one of the simplest models you could possibly imagine? A simple model will be a linear regression, a multiple linear regression. So I'm going to model Y as some mean, And then I'm just going to add up the effects of all the different SNPs. So there's my genotype, coded 0, 1, 2 perhaps again, and then the effect of that genotype on the SNP, beta J. So if I'm going to do that by traditional means with a linear regression, multiple linear regression, there's a very standard little formula. I'm going to put all of the genetic information into a matrix and then just do G transpose G inverse times G transpose Y to get beta hat. Very simple, but it doesn't work if P is greater than N. That matrix can't be inverted, it's singular. And even if P is not greater than N, but if P is close to N, I'm going to have perhaps some numerical problems doing this. So why does polygenic risk score work? Polygenic risk scores have not estimated things this way. Basically, they've just added up the results from each of the loci separately. Well, it works because if the different SNPs are uncorrelated, then that big matrix of G transpose G is actually diagonal. And therefore, when you invert it, you just end up with a diagonal matrix again. And that just means that you can simply add up the effects of all the uncorrelated SNPs. And then you can also get information from multiple sources. You can get information from very, very large studies, very large cohorts, or you can use meta-analysis to combine information across cohorts and estimate these individual locus contributions beta J extremely precisely. I'm going to say that it is possible sometimes to estimate them directly, basically with this sort of multiple regression model. But you can often do more precise estimates of beta by, combining information from multiple sources and putting it all together. So I've given you a really hand-wavy definition here of building a polygenic risk score. I mean, your goal is to estimate basically the central equation here, the parameters in the central equation here. What is normally done is that these beta hats come from public resources, from meta-analyses, or perhaps from the repository at the Broad Institute, and people then multiply those estimated values of beta by their own genotypes to obtain a polygenic risk score. The details do matter. I haven't talked about them, but the details do matter. Which SNPs exactly are you going to include? We know we have highly correlated sets of SNPs in the genome. So if you have a highly correlated block, like this block in red here, you can only put one of the genetic variants from that block into your calculations for the polygenic risk score, because it's really just one signal. And if you put it in, if there are 8 SNPs in this block, if you incorporated that's 8 different SNPs into your polygenic risk score, you would be essentially counting that locus 8 times. So the details that are tricky in building polygenic risk scores is how to figure out which SNP you're going to keep in a highly correlated block. And which ones you're going to keep overall, sort of how are you going to decide which SNPs are you going to keep from a point of view of significance? You could run your polygenic risk score calculations keeping only the most significant SNPs, or you could be far more lenient and keep everything with a p-value of 0.1 or smaller. So these are the details that do matter. There are a number of techniques that have been proposed for selecting SNPs from correlated sets. This is not something that I'm focusing on today. I have given you some of the references here. I should say that at the very end of my presentation, I have 2 slides that contain a more detailed reference list. And so at the very end, I'll make sure that I can show those so that they will end up in the recording. Some of the techniques that can be used are pruning and thresholding, or conditional and joint multiple SNP analyses, or Bayesian methods. But basically, these are all methods that try and select from among highly correlated SNP sets to make sure that the score doesn't contain redundant signals. This figure here comes from a paper by Kara et al. In 2018, where they built a polygenic risk score for coronary artery disease. And they showed that the risk of CAD for individuals who had high values with the polygenic risk score versus low values with the polygenic risk score was really very large, and approaching the magnitude that one would see for a monogenic mutation. So here you have, on the left-hand panel, in panel A, you have just the values of the polygenic risk score. They're rather anonymous values, but it's been scaled, so they run between -4 and +4. And the people who are over here on the right are at the highest risk of CAD. And if you take this vertical line in dark blue, and you look at the small number of individuals who have scores above this dark blue line, they have a 5-fold risk higher of CAD versus the rest of the population. And this is definitely sort of a magnitude of risk that would be comparable to what has been previously identified for monogenic mutations. They have a couple of other lines here, of course, showing different cutoffs and what their risks are associated. In I'm just hunting for my mouse again. In panel C here over on the right, you can see the percentiles of the polygenic risk score on the x-axis and the prevalence of CAD on the y-axis. So as you can see on the right here, when you get into the very, very high values of the polygenic risk score, you're talking about subgroups of this population that may have a 10% risk of CAD, which is really very high. So polygenic risk scores, if I make my second point, a very simple calculation, they can successfully identify high and low risk individuals. They are aggregating information from many, many SNPs across the genome, and so the PRS in itself doesn't necessarily come with any understanding of etiology. You don't need an understanding of etiology to build a PRS. It's just a number, it's just a score. It allows you to say this person has a high PRS and probably a high risk of disease. That person has a low PRS and a lower risk of disease. I'm going to pause briefly here again. Any questions at this point?
I do not see anything on the chat. I hope the chat is working for everyone. Should be.
Yep. Okay.
Yep.
All right. So now I'm going into the examples that I mentioned in my outline. So these are the work of some students in my group. Sahir Bhatnagar graduated maybe about 3 years ago now. Vince Vergetta is actually the research associate of a colleague of mine, Dr. Brent Richards. And so I'm going to talk about PRS scores in a number of different contexts and how they can be used. So here, we were building a PRS for bone mineral density using UK Biobank. Instead of building that PRS with the method I've just briefly described, where you basically just add up the results from a whole series of SNPs, independent SNPs, what we did here is we used a lasso model, so a penalized regression model. So the question we were trying to ask is how much variability in bone mineral density, BMD, is explained by genetics. So in the most clinical situations, BMD is measured by a DEXA scan and X-ray. Some of you may perhaps have actually undergone that test, where they scan your spine and your hips and whatnot with an X-ray machine. UK Biobank did measure bone mineral density, but it didn't measure it with DEXA, which would have really been too expensive and difficult for them to do with the number of individuals in that study. So they used a heel ultrasound measure instead. Speed of sound, which I'll refer to here as SOS. It's also sometimes in some slides, because of course they've been developed at different points in time, called EBMD, for reasons that I actually can't quite remember now why we did that. But so EBMD and SOS are sort of synonyms for this ultrasound measure of bone density in the heel. So if you look at this little graph here, the little histogram at the bottom, and the y-axis here is how much of bone mineral density, how much of the heritability of bone mineral density can we explain? How much of the variance can we explain? If you just look at standard factors like age, sex, and body mass index, or all of the risk factors that are standardly asked if you visit the doctor, so have you previously taken corticosteroids, etc., you will not explain very much of your bone mineral density variance. If you start to add in the genetic loci that were identified through genome-wide association studies, these are the two bars that are indicated with DEXA. It starts to come up a little bit if you use the results of a study of 30,000 individuals published in 2015. We can explain a little bit more than 5% of the variability in bone mineral density. If you do a GWAS, larger GWAS studies, now using UK Biobank data, but the earlier releases, now we're explaining 10% or even 20% in this 2018 paper of bone mineral density variance using a standard GWAS style approach and taking the significant loci, okay? The red bar here is the theoretical maximum heritability, not calculated from the genetic data, but estimated from family studies and that kind of thing. So I had said earlier that you can't estimate the model parameters from a linear regression when p is greater than n. Well, of course, that's not entirely true because we could use some of the many techniques that are available for doing estimation when p is greater than n. And the lasso is one of those. We couldn't do it with a standard linear regression, but we can do this kind of estimation with a penalized regression. So this is essentially the same equation I showed you before, but just flipped around. So I have y minus what was on the right-hand side. And then I add a penalty term to force the coefficients to be a little bit smaller. If lambda, I'm going to minimize this quantity. If lambda is 0, I would have a standard multiple regression. As lambda gets larger, what I end up doing is forcing all of those beta coefficients to be smaller than they might be if it was not penalized. This is an estimation procedure that you can run when p is greater than n. So given that UK Biobank provided an opportunity, with a really large sample size, we just thought we would try out the lasso to predict bone mineral density. Here's a far too complicated slide. Point being, first of all, that there's a publication that I have given you the full citation for at the end of the presentation today. Another point is that we collected individuals from UK Biobank that had the speed of sound measurements available, that had good quality control on their genotypes and were of white British ancestry. And then we divided the data into 3 sets: a training set, a small set that we've called a model selection set, where we're going to try and select the appropriate value of that parameter lambda, and then a test set. And so we're going to fit the lasso model. We have to decide which SNPs are going to go in, because even with a lasso model, we can't put all of the millions of SNPs into this model without it crashing and dying. So we did put a p-value threshold in. So then we're going to try a few different p-value thresholds and a few different values for the penalization parameter and optimize that here in orange. And then we're going to test how well the whole thing performs in the test set from UK Biobank, and also in a number of other data sets that were not included in UK Biobank. This is a slide of preliminary results that I just really like, so I've kept it here, even though it didn't end up in the the sort of final version of the paper. I had showed you earlier that if you just look at age, sex, body mass index, these are things that are known to be very predictive of your bone mineral density, and they explain about 5%, and I had showed that earlier in a different kind of graphic. If you calculate a polygenic risk score in a traditional way, so you do pruning and thresholding, for example, and then you take different p-value thresholds and you calculate your polygenic risk score, you will certainly do much better than simply age, sex, and body mass index. You will explain something more like 15 or 16 or 17% of the variance in bone mineral density. However, if you use a lasso, which is here referred to as a machine learning algorithm, you make a substantial jump again. So now the variance that we can explain in the bone mineral density has jumped up to close to 25%. So what was that built on? That was built on a GWAS of speed of sound in the UK Biobank data. So here is an even more modern Manhattan plot for bone mineral density, which comes from this 2020 publication. So you can see there's many more loci than there were in the 2009 publication on bone mineral density. And here are some of the lasso tuning parameter results. So what happens when you run a lasso is as you change the value of lambda, which is on the x-axis in these plots, some of the estimated values for the contributions of the SNPs, so the beta values, their estimated beta values, are forced to be 0. So here's beta hat on the y-axis. I know it's rather small, but the center line is 0. And as you vary, I'm sorry about that. As you vary lambda, you will have all your coefficients being forced to 0 on the left-hand side, or none of them being forced to 0 on the right-hand side. And what you're trying to do is figure out where is the optimal value of that penalty parameter. And using cross-validation, the plots here with the black, my mouse sometimes disappears. There it is. The plots here with the black dots are showing you which is going to be the best value of lambda. And we're choosing the value here that corresponds to the minimum mean squared error. The different panels on this graph are corresponding to different p-value thresholds. So here, this is 5 times 10 to the minus 8. We would only be including in the lasso model SNPs that in the individual GWAS were significant at 5 times 10 to the minus 8 or more significant. And then I have 4 graphs shown here. I have 5 times 10 to the minus 8, 5 times 10 to the minus 7, minus 6, minus 5. Each time I can get a different lasso model. Each time I may get a slightly different optimal value of the penalty parameter. So that's we picked the best of all these options. Now, if I go back here to the plot that I showed you before on how much of the variability in bone mineral density you can explain with various models, I've added a bar. I've added the yellow bar on the right here. So this is coming out of a lasso model, and it's a little bit better than what we had with the green, which would be a standard sort of PRS approach. So we're getting a little bit better performance, or we'll be able to capture a little bit more of the variability in bone mineral density by using the lasso model. So here's my point number 3. that alternative estimation methods can improve PRS performance. There is an important caveat here, which the benefits or the potential improvement seems to depend on the architecture, the genetic architecture of the trait. If you have a monogenic trait, you're not going to gain anything by doing this. If you have a trait that is only has a few loci oligogenic, you probably also won't gain anything here. And if it's omnigenic, again, you may not gain anything because we did have to apply some p-value filters in order to even fit the lasso models. I should say that it wasn't easy to fit those lasso models, just computationally. We ended up using the Amazon cloud because our own clusters were incapable of holding the matrices of the size that was required. So somewhere in the polygenic architecture space, doing some new modeling techniques may be able to provide potential advantages. I only caught the last few minutes of your previous speaker, but it seems to me that they were talking about alternative estimation techniques and where they have benefit. And I think a point to add to that is, well, what's the genetic architecture? And there will be situations where you may possibly get more benefit and other situations where you may not be able to get much more.
Can I ask?
Yes,
yes. Yeah. So because you showed that the example of age and BMI on their own not doing very well in terms of risk prediction, but there are indeed other settings, right, where non-genetic factors are very important, often like smoking and things like that. And you see in the literature an increasing number of papers, I guess, where they are taking the PRS score and let it interact with such non-genetic factors. Now, what is your take on that? You know, having the PRS score interact with these non-genetic factors, or rather doing a GWAS, you know, in a gene environment interaction study and then deriving a score from it? Is there any, you know, evolution in that field?
I want to make a couple of comments. One of them here, this blue bar, the second bar, is all Frax risk factors. And for anybody who works in osteoporosis, those are what are usually asked in the clinic. Okay, it doesn't do very well. So that includes age and sex and body mass index, but it also includes those other things. Well, not smoking, I don't think, but it includes everything that is standardly asked when you go to the doctor and get assessed for risk. That's one minor point I wanted to make. The other point I want to make, I may defer and come back to, because I do speak more about that later in this talk today. I think it's a really interesting question, and I don't think we have all the answers yet, because we have been trying. So that's why I'm actually going to talk a little bit about it later, and maybe we can come back to that discussion.
And what's your take on neural networks, etc.? I mean, it does indeed depend on the underlying genetic architecture, which is largely unknown. But yeah,
we tried neural networks. We tried gradient descent methods. We tried a whole series. In this example, the bone mineral density example, my colleague, Dr. Richards, he's an endocrinologist, and this is the trait he works on a lot. And they did not do well. No, the lasso did better. Yeah,
SNP data is not the the most ideal data for neural networks, but it seems that if you set up some simulation studies where there are interaction patterns or really complicated structure, which of course we also cannot prove, then they do dramatically better, of course, than the linear methodologies.
Absolutely. Yeah, you need interactions to be there. The question is, how big are those interactions, Because if the interactions are very small, you won't gain much. Yeah. Yeah. I think these are questions that need to be answered over the next couple of years. Yeah.
Yeah. I don't see other chats.
Okay. All right. So I'm going to, excuse me. I'll move on to my next topic. What can we do with polygenic risk scores? And I'm sure you're seeing a trend here. Everything is talking about osteoporosis and fracture. So here's a graph of basically a polygenic risk score on the x-axis here, and it's divided into in deciles, and the risk of hip fracture on the bottom or the risk of major osteoporotic fracture on the top is clearly showing to track with the risk score, the polygenic risk score for bone mineral density. It's probably not a big surprise. I mean, if your bone density is low, your risk of fracture is high. But here we see that the score is actually built for the bone density, and it tracks very nicely with fracture risk. And this is true at all ages. We looked at it in all ages. This is measured with the ultrasound speed of sound, as I mentioned before, from UK Biobank. If you actually measure it with DEXA, it is going to be more accurate, but it still does a relatively decent job. Now, here is what can I do with that? This is a rather complicated slide showing sort of the standard practice right now for preventing major osteoporotic fracture in the elderly. So here's your population of individuals. If somebody is very young, you usually say don't worry about it. If they have a risk factor, or if they're over women, if they're women who are over a certain age, they might end up going for a DEXA scan, right? So you might first do a little bit of a calculation to see if they seem to be at risk. If they are very high risk, they're going to be perhaps treated. If they're very low risk, they won't. If they're in a moderate risk category, they might be recommended to have a DEXA screen. And if the DEXA screen looks bad, then they're going to be headed off for treatment or reassured. Okay, so this would be a sort of standard practice at the moment. Now, what if you had a polygenic risk score? What would you do with it in a population screening context? Well, this is a hypothetical, a thought experiment of what you could possibly do. You could say, okay, I have my population that are over 50 and have at least one risk factor. I could then do some genotyping on them and then calculate their PRS using my GWAS measure. And if it's high, as in I predict that they have high bone mineral density, I'm going to put them over here into the reassurance category. I don't need to worry about them. I have predicted that their bone mineral density will be high. That means that they're unlikely to have a major osteoporotic fracture. But if the GWAS is not high, then they're going to continue through my flow chart that would be standard practice in the clinic. And if they, we would send them for a DEXA X-ray scan if that seemed to be appropriate. So what do we have? We have the standard approach where we might recommend an intervention to the people who ended up on the red side of the diagram, and we would reassure those who ended up on the green side of the diagram. And then we have a sort of thought experiment on what might happen if you modified screening. And again, individuals will be classified as recommending an intervention or reassuring. And we could say that a true positive is somebody who we recommended intervention to, who really needed to have that, etc. So we can define sensitivity and specificity based on what actually happened to these individuals. So we did that thought experiment using UK Biobank data. And this is what comes out. It's a bit of a complicated slide, so I'll try and walk you through this a little bit. So we have in blue standard practice, screening strategies without including any genetic risk scores of any kind. And in red, the modified thought experiment of a clinical process for screening that includes a polygenic risk score incorporated into it. The first thing is take a look at the second last pair of bars, subjects having CRF fracs but reassured. So the red bar is a lot lower than the blue bar. We have reassured people. Their PRS was high, suggesting that they are unlikely to have a fracture, that their predicted bone mineral density is high. And so there are more individuals that are sort of taken out of the process here that don't need to go down further in the flowchart. We have a reduction in the number of people who actually need to have the X-ray based screening of their bones. So that's a healthcare savings potential. So those are the good things. What's the pain? What are we paying for that? Well, we are seeing a slight drop in sensitivity. So we are perhaps missing a few individuals who probably should have had a DEXA screening and maybe were at high risk of a fracture. And we're gaining a little bit of specificity through this process. So if you think about it at a healthcare setting framework, not at the individual person, there are potentially quite substantial cost savings associated with not sending people for DEXA screening who don't need to be sent for DEXA screening. The price you're paying to do that is that you have a slight drop in sensitivity. There are some individuals you've missed. So like I said, this is a thought experiment. But I think it certainly opens the door to thinking about how you might incorporate PRS into population screenings. And so here's my point number 4. The PRS may be helpful and in terms of trying to optimize screening at the population level. Okay. I could take a question there if there is one. Otherwise, I'm going to move on to my next topic.
But to validate, you then need to be sure that you are working with the same population, right? And that all these aspects of heterogeneity are taken care of, right?
Oh,
how much
yes.
of a concern is that when you would like to translate it into clinical practice?
That's going to be a large concern because all of that work was done in the British subset of the UK Biobank. Yeah, absolutely. So you would need, if you were going to do something like that, you would need to have effective scores for different ancestral groups. Otherwise, it would be very dangerous to apply. Mm-hmm. Yeah, I agree. So here's another topic, common versus rare genetic variants. So we know PRS captures signals from genetic variants with significance in GWAS studies. So these are going to be variants that are seen in multiple individuals. They won't be the kind of mutation that you've seen only once because it would never then show up as significant in a GWAS. And that means it really excludes most rare pathogenic mutations. They're not incorporated into going into the calculation of a polygenic risk score. However, given that you have somebody in front of you, perhaps in a clinical context, who has a disease, what's the probability that they do carry a rare pathogenic mutation? So I'm going to write this mathematically, or quasi-mathematically, the probability that somebody carries a pathogenic mutation, given that they have the disease. They're sort of in front of me in a clinical context. And I want to refine this and put the PRS score in there, assuming I had it, assuming I had that PRS. So now I want to figure out what's the probability that somebody carries a mutation given that they have a disease and that I have a polygenic risk score for that individual for that disease. So now just doing a little bit of manipulation of probabilities using Bayes' rule. I can flip those probabilities. This is the one I had on the previous slide. I can flip that around to be the product of 3 other terms. What are these things? One of them on the top here is the probability that that person has the disease, D, given that they carry a rare pathogenic mutation, M, and that they have a PRS score. So we're going to make some assumptions here. The first one is that if they carry a rare pathogenic mutation, M, that the PRS probably doesn't make much difference on top of that. Okay, that's an assumption, but that's the assumption I'm going to work with here. Second assumption, the probability of disease, given that I have a PRS score, I'm going to assume it's been estimated already by some other study. The third thing I need is what's the probability of carrying a rare pathogenic mutation, given that I know the polygenic risk score value. So this is what I'm going to explore in the next slide. That's why I've put 2 question marks here, because it's coming up. This is the work of a PhD student, Tianwan Liu. So we took exome sequencing data from the UK Biobank. So one of the earlier releases, 44,000 individuals who were at exome sequencing. We looked at 5 diseases for which rare pathogenic mutations have been reported. And we looked at the actionable mutations following the American College of Medical Genetics and Genomics. And we added a few more there, a few validated rare dominant mutations. And we calculated polygenic risk scores for these 5 diseases following, well-cited publications with the coefficients that had been included. So here's 2 of the diseases just to illustrate the kinds of results that we were getting. So what is the odds ratio for being a carrier of a breast cancer mutation, pathogenic mutations here? So here are the individuals diagnosed or undiagnosed with breast cancer, who are carriers or non-carriers of a pathogenic mutation. So from the exome sequencing data, right, in the UK Biobank. So the odds ratio is quite large, 4.83. And we also have an odds ratio associated for a sort of a 1 unit increase in the polygenic risk score of 1.65 for carrying breast cancer. So here, what I'm doing here is looking at the 2 things separately. So I've got my breast cancer risk associated with the polygenic risk score, or in the bottom panel, bottom part of the table, I have the risk associated with colorectal cancer of the polygenic risk score. And then separately, I'm basically just looking at what's the risk of this disease if they carried a rare pathogenic mutation annotated from among the actionable mutations. So these odds ratios are not surprising. These are actionable mutations. They really should be putting people at higher risk of disease. Now here, the previous result is now divided by the polygenic risk score. So what we have is a polygenic risk score less than 50% on the left-hand side of the figure and greater than 50% on the right-hand side of the figure. So what is shown in each of the 4 panels here is that if your polygenic risk score is high, as in you have the PRS says you're at a high risk of the disease, there isn't very much difference in the prevalence. The prevalence is on the y-axis for individuals who do or do not carry a rare pathogenic mutation. So the colored dots are the individuals who carry the mutations. The gray dots are the people who don't have the disease. But on the other hand, if you go to the left-hand side, I now have a big difference between those who carry a mutation and have the disease, and those who don't. There's a weird interaction going on here, to say that sort of in a rather generic way, perhaps a rather weird interaction going on between the polygenic risk score and the risk of disease for people who do and do not carry rare pathogenic mutations. Plotting the same thing slightly differently, here's the PRS score on the x-axis. Here's the probability of carrying a rare actionable variant on the y-axis. Obviously, these data have been smoothed. If your polygenic risk score is small, the probability of carrying a rare actionable variant can be high. This is among individuals who have the disease. Among the individuals who have the disease. So obviously different diseases, red for breast cancer, yellow for colorectal cancer. I'm going to make my point and then come back a little bit further on the discussion. Diseased individuals with low polygenic risk scores are more likely to carry rare pathogenic mutations. This may seem strange, but it is actually an example of collider bias. So we have both rare and common genetic variation influencing the disease risk. In fact, these things are largely uncorrelated with each other because the rare ones are so rare, they're not going to be correlated with what's much of the common genetic mutation. But if we condition on who has the disease, we are actually inducing an association or forcing an association, and that's collider bias. So here's my collider. This is collider bias, this little schematic. I have some exposure, I have some outcome, and I have some collider. So what do I have? My exposure here in this context is my polygenic risk score. My outcome is who is a carrier of a pathogenic mutation, and the collider is the disease. If I condition on who has the disease, these two things will appear to be correlated even if they were not in the original full data set. So the artificial division of genetic risk into common and rare, because of course it's a bit of an artificial division. At what point do I call something common? At what point do I call something rare? It might be useful, or it can be useful, because maybe it could be used to prioritize people for sequencing. So I have somebody in front of me who has a disease. I calculate their polygenic risk score, and actually it turns out that they seem to be at very low risk of disease based on that polygenic risk score. That's coming from the common genetic information. But maybe that's going to say to me, maybe I should send this individual for exome sequencing. Maybe that individual is now carrying some rare pathogenic mutations. Maybe that would have implications for their family members and for counseling. So I could maybe play with this idea of common versus rare in a clinical context to prioritize individuals for screening of other kinds. Okay, I have one more story. Do we have time for it?
It depends how long.
I think I can do this one quite quickly.
Yeah, go ahead. Yeah.
Okay. So it's my last topic. This is the one you wanted. I said I was going to come back to, so I'm glad I have a few minutes for this. How much interpretation can we extract? I showed this figure already, as we know the PRS can confer risk. But here again, it's the same student, by the way, he's a really brilliant student, Tianwan Liu. Well, here I'm going to talk about diabetes, coronary artery disease, and polygenic risk scores. We know that the PRS for CAD works well. It's published by Kara and many others. Does it still work in subsets of individuals? So here we're going to ask very specifically, does this PRS work among individuals who have type 2 diabetes? Type 2 diabetics have a much higher risk of heart disease. We know that. We've known that forever. And can the PRS point to mechanisms? So in this 2020 paper in cardiovascular diabetology, we took 21,000 individuals from UK Biobank who had reported diabetes. And I'm just going to say diabetes rather than type 2 because it's a question of how accurate the question was posed, you know, in the UK Biobank. And I'm also going to take a tiny study from McGill with 352 individuals who, these ones definitely had type 2 diabetes, but all of these people had coronary angiograms. So these are just describing the two cohorts. Obviously, one's large, one's small. We have the standard risk factors we know, but we don't know about multi-vessel stenosis in UK Biobank because they didn't have angiograms. We do know about multi-vessel stenosis in the small little cohort because they all had angiograms. And we can look at associations for heart disease or we can look at in the two cohorts for associations with standard risk factors and heart disease. I mean, we used a polygenic risk score for heart disease, so it's strongly associated in UK Biobank. And we can look at associations with risk factors for heart disease. In both cohorts, this cohort's so small, we don't have a vast amount of power. But what we can do in this very small cohort is notice that the PRS for heart disease, we can't calculate that, but we can see that there's a very strong association between multi-vessel stenosis and the PRS in this particular cohort. So here we are looking at the association between the polygenic risk score and these other risk factors, okay? So is that polygenic risk score strongly associated with hypertension or lipidemia or poor glycemic controls? These are all risk factors that are known to be associated with heart disease. I mean, yes, in UK Biobank, they are. You know, the confidence intervals do not include 0 for hypertension or hyperlipidemia. They don't. The confidence intervals are wide in our tiny little cohort. Its sample size is obviously an issue. But these odds ratios are not huge. The family history one is larger, not surprisingly. But look at the size of this association. It's very large and dramatic. So here, you know, this is not by any means a definitive study, but it is a suggestive finding that among diabetics, the polygenic risk score is picking up the risk of multi-vessel stenosis, and that may be the mechanism by which individuals with diabetes have that elevated risk of heart disease. It doesn't seem to be drastically coming through hypertension or hyperlipidemia. This seems to be perhaps the mechanism that puts those diabetics at risk of heart disease. I realize it's hypothetical. So 2 more points here. The association between the coronary heart disease PRS and multi-vessel stenosis points to a particular mechanism. And point number 8, PRS associations in subgroups may differ. So given that I have definitely talked for my 75 minutes, I'm going to skip a couple of slides on population transfer. We know this is a big caveat. PRS do not transfer well across populations, and an area that definitely requires a lot more work. And here I've just simply summarized all the points I've made during the talk, and now can pose to you, where now? And any questions?
Thank you very much. It was a very comprehensive story. Yeah. Are there questions from the audience?
I know I promised you references. I'll make a plug for our PhD program. I will show some acknowledgements, and I'm just going to, where are those references? Show the reference slides so that they end up into the recording. There's 2 pages. Here's the first one. Here's the second one.
Okay, Martha, the PRS are taking also into account the inner differences that are present in a single population, so basically within population heterogeneity. Are they taking that into account? That was the question.
They will be averaging it out. You know, a PRS within a single population, we have to make the assumption that if there are differences within a population, that they aren't too large or too dramatic, but they're, of course, we know that allele frequencies vary slightly, even north to south of the UK, for example, that's been well shown, or west to east in the UK. So that will slightly change a PRS score. If it's those kinds of things will probably reduce the accuracy of that PRS and attenuate its performance a little bit. It's still probably a very useful tool. The dangers are if you try and take them across populations, because then the differences in the genetic architecture tend to be much, much larger.
It's also
a question. Come back a little bit to your question, Christelle, about interactions with PRS and other factors, and or looking at subgroups of individuals. So, like I said, I did the study with the diabetics, but you could also look at individuals who are I don't know, living in a particular environment. I think we have a little bit of distance to go there before the results start to be stable. We've played with this, we've tried. We get results that I don't consider to be stable. As soon as you start subdividing, you have smaller sample sizes.
Yeah.
And so you have to be a little bit cautious to make sure that the results you have really are reproducible, and you're going to get the same result if you did the same thing in another study in another place. I think it's actually one of the exciting areas to explore right now, but I think it has to be done with a lot of care.
Yeah, and maybe also to come back to the previous question, so it's always an issue of how much heterogeneity do you allow? I mean, you can quantify that mathematically, but it doesn't necessarily translate to also instability at a PRS score that is still biologically interpretable. And
yes. I mean, there's obviously different things going into that PRS calculation, as simple as it is. One of them is the allele frequencies of the different variants, and the other is the beta hat, the estimated contribution of that variant. If that beta hat is not the same in some exposures versus other exposures, in some environments versus other environments, then your PRS is wrong in some way, right? You actually would like to have exposure-specific estimates of beta hat. But how do you get those exposure-specific estimates? You need really large sample sizes to get them done with any accuracy. So you have to be very careful.
Yeah. We also have a question from Benno. He's the local organizer. So given two PRS scores, are there meaningful interpretations of the overlap between gene sets that might be constructed to correspond to these two scores?
I mean, I think the answer is yes, for sure. I haven't talked much at all about interpretation of PRS scores because it's it's so tricky. You have thousands and thousands of variants going into the score. Normally, you haven't placed any constraints, biological-based constraints or pathway-based constraints on the construction. So going to interpretation of what you end up with is not going to be straightforward. I mean, the example I presented with the diabetic patients and the multi-vessel stenosis is one way to look at interpretation. That's a bit of a secondary way. We haven't opened the box on the PRS. We're just trying to figure out what it does and what it's most associated with. If you want to go into interpretation of gene sets, then you have to link your SNPs to genes. If your two PRS were constructed in different populations, they may have selected different SNPs from a correlated set. You have a little LD block, maybe study 1 picked SNP 1 and study 2 picked SNP 2. So to do the interpretation, you have to realign those SNPs back to the LD blocks, look at interpopulation differences, and then get to interpretation. So it's tricky. It's very tricky to actually do. That doesn't mean it shouldn't be done or can't be done, but yes, it's not easy.
Mm-hmm. Yeah, thank you. Once more, thank you very much for the interesting talk, and well, hopefully we see each other at some point in person and can go for a coffee or tea. Yeah. Yeah.
Thank you very much for the invitation to join you, and I hope the rest of your school or your week is very enjoyable for you all.
Okay, so it's my pleasure to introduce to you Kimberly Glass. I've had a pleasure to meet her have met her already a few times in Boston on individual specific networks, and you may have seen when you had a look at our website that several, well, at least one topic of our ESRs is on individual specific networks, but there are several other people who are triggered by personalized networks. Now, Kimberly's group is part of the Systems Pathobiology unit at the Channing Division of Network Medicine at Brigham and Women's, which is a teaching hospital at Harvard Medical School. But her primary interest is in methods development, and she's particularly interested in integrating multi-omics and using network theory for that. But apart from methods development, she also spends quite some time on developing the tools, and she's creating an entire zoo of tools and algorithms. And a monster, anaconda, I think, as well, or well, a lot of animals. And I'm sure that she will identify many more animals to populate that zoo. So I give the floor to you, Kimberly, and we are very much looking forward to your talk.
Excellent. Well, thank you, Crystal, for that introduction. I know that it's late in the day there, so I'll do my best. For those of you who hanged on to the very end, thank you. I'll do my best to make it worth your while and also not eat in too much more into your evening as it gets dim out. So as Crystal said, I am primarily a methods developer. My background was in quantitative science. I got my PhD in physics, but now I primarily do multi-omics and computational biology in the context of network medicine. And I often like to set the stage for my research by telling what I feel like is a fairly simple story and one that we often run into in the clinic. And that is the story of two girls. I named them Susan and Jane here, but they could be any of your two favorite names. And these two children grew up down the street from each other. They were friends, they played together, they had a really good life. Over time, they grew up and they developed habits. You know, Susan occasionally liked to eat a really good hamburger. You know, that red meat was extra tasty. For Jane, she liked to have her glass of red wine every evening. Over time, as we all do, they grew up and they got older. And eventually they got sick and they came to the doctor and they had very, very similar physical phenotypic manifestations of their disease. So the doctor treated them with the same drug, right? He gave them the same treatment because they looked similar. And something interesting happened. Susan got better, but Jane didn't. Now, I would like to argue that this is part of what could have been driving this difference between Susan and Jane. When Susan and Jane were young, the underlying network in their cells were both healthy, slightly different. They have different genetics, you know, different ancestry, but you know, overall a healthy network. Then over time, through both environmental exposures and also their predisposition in their genetic profile, their networks changed. And what happened is the network became unhealthy. But it became unhealthy in a way where Susan's we know how to treat. We're targeting the right area of the network, but Jane's we didn't. And that leads me to the importance of biological networks. Why do we even want to study this notion of how, you know, the underlying cellular network? Well, the reason why my group studies biological networks is because of this idea of biological processes. We know that a gene, a phenotype is not driven by a single gene, a single protein, a single genetic variant, but by connections linking them together, by the processes in the cell. One type of network that I am particularly interested in is the gene regulatory network. And the gene regulatory network, there's kind of a toroid diagram over here on the right. You can think of the nodes being the genes and the edges being relationships between the genes. And in particular, the edges are directed to indicate causality going from transcription factors to their downstream target genes. And most importantly, as illustrated by that story I just told, By comparing these regulatory networks, we may be able to gain insights into alterations in the biological processes that can underline differences in disease or phenotypic states. Now, my background is quantitative, it's not biology. So when I try to think about gene regulation, I tend to start with a very, very simple picture of a line and an idea of where the gene starts along the line, where the line is the DNA sequence, and this arrow here represents the transcriptional start site, or where the gene starts in the genome. So how does gene regulation work? Well, there are many different mechanisms that contribute to gene regulation. The primary one that people often think about is transcription factor binding. And there are two main ways that we can measure transcription factor binding sites. One is through in vivo methods, such as chip sequencing, chip data, where we can find the actual locations transcription factor bind in the genome. The other is more computational, where we can do something like a transcription factor motif scan along the DNA sequence to find potential transcription factor binding sites, where the DNA sequence matches basically the DNA binding domain of the transcription factor. What else contributes to gene regulation? Well, of course, the transcription factors themselves don't act in isolation. They can form complexes. It could be that this transcription factor, this red one right here, can bind directly to the DNA, but the blue one then binds to the red one through a protein-protein interaction, and they work together to regulate gene regulation. That's not the end of the story. Along the genome, as we heard in our previous talk, you have genetic variants, you have SNPs that can impact where transcription factors can bind and how proteins are translated and all sorts of other things. You also have CG sites, CPG methylation sites, where you can actually have little methylation units poking out of the DNA that can impact these regulatory mechanisms. You have three-dimensional structure of the genome, right? You have things like chromatin state and 3D chromatin structure. And then finally, at the end of this process, you have an mRNA that comes out of the gene. That's illustrated by this little wiggly line. But the mRNA isn't the end of the story, right? There can be things like microRNAs, post-transcriptional modifications, where microRNAs come and bind the mRNA and degrade it. And then ultimately, you have protein expression. So that's a very, very complicated story. And I'm trying to argue that what we really need is this. We need to understand the regulatory process, how transcription factors come and regulate genes and basically cause them to manifest into biological function. So how does this work? Well, we know, quote unquote, there's an underlying regulatory network, just like we know there's a social network, just like we know that there's a network of connections on the internet. We know there's something, but we can't directly measure that regulatory network. What we can measure is we can measure different types of omics data, the different types of data I just described. And we can use those to try to infer the regulatory network. Now, unfortunately, this is an incredibly complex problem. Didn't already believe it, hopefully, after that last illustration, you really do. You know, you could argue that it's really important to understand transcription factor regulation. Maybe genetic variants are the key. Maybe it's the way the chromatin is folded around. Maybe it's the CGs, right? It's kind of like the story of the blind man and the elephant. We're all feeling different sides of this regulatory process. So in my research, the three main mechanisms I tend to focus on are the follows, and I'll get into some of the others soon, but I'm going to kind of give you a crash course on these three mechanisms that kind of form the heart of the methodology I developed. I'd like to start with the idea of transcription factors. We know that these are regulatory proteins that can do something to mediate gene expression. So transcription factors do not act in isolation. Transcription factors come together and they form protein complexes. We know how to measure these protein complexes through things like protein-protein interaction information. There's lots of databases out there, and there are also assays you can use to understand how proteins come together and form these complexes. These transcription factors then bind upstream the gene in a region called the promoter region. These we can also measure. These are protein DNA interactions. In that previous illustration I showed, these are things you can get through things like scanning the DNA for transcription factor binding sites or looking at chip data. These transcription factors then recruit RNA polymerase. RNA polymerase slides along the gene, not quite that simple, but more or less, and this produces an mRNA product. And this is what we think of as gene expression. This mRNA is then translated into a protein. This protein might be a transcription factor that creates another complex, and this whole process repeats. But what I'd like to kind of emphasize here is that these are three different types of biological mechanisms that we can measure using three different types of data, but they all represent the same underlying process, namely the transcription factor regulation of genes. So how can we use this to build gene regulatory networks? Well, about 8 years ago now, I guess, the time just does fly, I developed the first zoo animal, as Crystal mentioned, that is PANDA, which stands for Passing Attributes Between Networks for Data Assimilation. And it takes these three sources of input data, protein-protein interactions, protein-DNA interactions, and gene expression, and casts them into three basic network models, namely cooperation between pairs of transcription factor proteins, whether or not those can form a complex, potential regulatory events from transcription factors to downstream target genes, and then potential co-regulatory events, whether or not two genes are co-expressed can be used as a proxy whether or not they're co-regulated. PANDA then builds what we call a multi-layer or multi-tier network using these three sources of data, basically drawing connections between pairs of transcription factors if we know that they can form a pair or in a same complex. Interaction between transcription factors and their downstream target gene, if, for example, a potential binding site for the transcription factors from the promoter region of the gene. And then interactions between pairs of genes if those genes are highly co-expressed, i.e. highly co-regulated. PANDA then applies what's called a message passing approach to basically say these three sources of data represent the same information. So let's get rid of inconsistent structures and emphasize shared structures to basically refine the information, to prune things and refine them to get a more complete picture of what's going on. And that does that through calculating two measures, responsibility and availability, and passing information back and forth this way. So very briefly, I won't get into the math in too terribly much detail, but I wanted to give you a sense of how it worked. How does PANDA actually integrate this information? Well, as a framework for the gene regulatory network, PANDA starts with a map from transcription factors to gene, which is based on the transcription factor motif scan, whether or not the transcription factor binding site is located upstream of a given gene. It uses this to construct an initial gene regulatory network. It then adds in information from the protein-protein interactions and gene-gene co-expression or co-regulation. As I said, this is co-expression and PPI data. And it then uses that to calculate the responsibility and availability. And essentially what happens with the responsibility is we say if two transcription factors target the same gene, or we think they do, and those two transcription factors are known to be a protein complex, we think that the edges between those transcription factors and gene are more likely to be correct. So let's upweight that. Similarly, if two genes are co-expressed and they're targeted by the same upstream transcription factor, we think the edge from that transcription factor to genes is more likely to be correct because there's consistent information, right? And so that's kind of two sides to the same coin. It's the same way of looking at the data. So that's the responsibility and availability. Basically upweight edges with consistent structure and downweight those that don't have it. You use that to update the initial regulatory network, and you basically flip the equation on its head, right? You say that if the same pair of transcription factors are known to regulate all the same genes, maybe we missed the protein-protein interaction. Let's update the PPI information. Similarly, if a bunch of genes are co-regulated by the same set of transcription factors, maybe they should be high in this co-regulation matrix, and we missed the co-expression due to all sorts of biological or technical information. This process then repeats to basically emphasize the shared information. So that's how PANDA works. Since its publication in 2013, we've applied in all sorts of situations. Because I wanted this talk to have more of a medicine focus, I'm going to briefly highlight 3 papers that emphasize and illustrate how it can be applied to gain insights into disease and even potential treatments. I will say before I get into these that these are all complete papers on their own. I'm going to have only 1 or 2 slides on each. So if you have any questions, please feel free to follow up or read the papers in full. So the first application I wanted to highlight was an application in sexual dimorphism and chronic obstructive pulmonary disease. And very briefly, COPD, chronic obstructive pulmonary disease, has traditionally or historically been associated with men, but that was because historically men smoke more than women. Once women started smoking at more equivalent rates than men, it was realized that when women get COPD, they tend to get it younger and a more severe disease course. So there's clearly sexual dimorphism in how the disease manifests. In the case of what we did, we decided we wanted to see if this was due to differences in the underlying gene regulatory network. So I use data from the Eclipse cohort. I forget what that stands for, something COPD. And we were able to build gene regulatory networks with Panda for both blood and sputum separately in both men and women, males and females. And this is important because the sputum is something that is deep down in the lung. It's probably more disease relevant. And the blood we treated a little bit like a control. Even though it came from a disease population, we knew it was more distally located from where the disease was manifesting. We then compared these networks between males and females, as well as between the blood and the sputum, to identify sex-specific network edges and those that were specific in the sputum, which were actually more disease relevant. And this is the subnetwork we were able to identify. And in particular, I wanted to draw your attention to 3 transcription factors of note. First off, I'll talk to you about zinc finger X. Well, as you can tell, zinc finger X has an X in the name. If you dig into what it is, it's an X-linked transcription factor. Something that is X-linked, you could definitely imagine having some sort of sexual dimorphic properties. Why it showed up in relation to COPD, we don't know. But it was definitely a little bit of a reassurance that the fact that it had different sets of edges in females and males made a lot of sense. The second one I wanted to highlight is SOX5. So SOX5 came up in our model, and it had a bunch of edges only in men. Interestingly, when we dug into the literature about this particular gene, it had been identified as a potential risk factor gene for COPD. It hadn't been highly studied, but it had been identified that way. And so one thing that might be true was that it was identified, but it hasn't had a strong signal because it may actually only be important in men and not women. And it's something that, you know, it would be very interesting, in my opinion, to follow up. And then the last one I wanted to highlight is the big ball right here around USF1. USF1 is known to dimerize with the estrogen receptor protein. And I believe it also is targeted or has a binding site for ESR1, right?
Kimberly, we don't see a ball. We don't see a ball. Is that no, we just see the 2 bullet points and then the 2 pictures on the left, women, men, and yeah.
Oh, this is very unfortunate. Can you see it now?
We see just no treatment and then dexamethasone. Now we see an.
Okay, let me pause. It may just be delayed.
Mm-hmm.
Please let me know if you see a network.
No, not yet.
What are you looking at right now?
Application with the 2 bullet points, and to the left, women, men, colored slide, and the comparison of 2 networks.
Interesting.
And then a white area.
White space.
Yeah.
Yeah, the network was supposed to appear. I'm going to hit forward one more time. What do you see now?
A slide application and then just titles, no treatment and dexamethasone.
So I don't know why you can't see the network picture.
It needs to cross the channel.
I don't know why. There is a network picture that has a huge hub around a particular protein, USF1. So I don't know how to get it to show up because it's on my screen. I apologize. Can you at least see the reference in the bottom left? Down here?
Yes, we can, yeah.
Right. So if you're interested in knowing what the network looks like, you can look in that paper. I don't know why it's not showing up on the screen. But yes, there's a huge ball there. I think I'm going to move on just because I don't know how to get it to show. I apologize to everyone. I don't know why it's not showing. If we were in person, you would see it on my screen.
Yeah. Could you share with us the slides as well? I mean, they are recorded anyway, so then we can send them around to the participants. Yeah. Okay.
Of course, I'd be very, very happy to. All right, and please interrupt me if anything isn't consistent again, so that I can talk through what it actually looks like better. All right, so the next application I want to talk about is looking at the effect of treatment in asthma in the context of regulatory networks. And in this case, we had sets of children who had been given either dexamethasone as a treatment or a sham treatment, essentially a saltwater treatment. And some responded and some didn't. Can you see a network picture now? When I click forward?
Yes, a purple one.
Excellent. Okay. Okay, so this is the network. I'm glad you can at least see this network picture. There's lots of network pictures in here of the network on the children who were given a sham treatment, the control treatment. And the network in red, if it shows up, for those who are given the dexamethasone treatment. And if we tab back and forth, again, I hope that this is working on your end, and if not, I will share the slides. You'll see that there's subtle differences in these networks. And what we were able to do in this particular paper was we identified hubs in these networks that were changing their targets between the individuals that had been given a sham treatment and that were treated with dexamethasone. And in this particular paper, we had a huge advantage. The expression data we used was based on cell lines that had been immortalized from these children, right? So they knew whether or not they were going to respond. We knew whether or not they'd been given a treatment, and we also had their actual original cell lines associated with them. And we were able to go in and perform siRNA knockdown of some of these transcription factors and actually show how they impacted downstream gene targets. And if you're interested in that particular type of network medicine application, I would encourage you to read this, this paper here noted in the lower left. The final paper that I wanted to touch on is about differences in subtypes in ovarian cancer and how we use PANDA to characterize differences in the subtypes. And there should be another network picture on here. Again, if you can't see it, I apologize. And it should be a network picture with many, many red edges. And in this particular application, we are looking at two subtypes of ovarian cancer. One which was angiogenic, which had already started forming blood vessels, and one which was non-angiogenic, one that had not gotten as far along in the cancerous process and had not started forming blood vessels yet. And we built these two networks and we compared them. And again, I don't know if you can see the network or the animation, but here are the two networks laid on top of each other. The only thing changing between these two are the edges. And the edges change, but it's really, really hard to see differences between these networks. And then here are the two networks together, the blue and red edges. And so the first thing that we asked when we saw this picture was, how in the world can we interpret this information? The last two pictures I showed you, we at least had a small enough network. We could just look at hubs. We could just say, okay, let's do some knockdowns of this. But in this particular network, it was really hard to figure out how to even interpret this. And this is your typical hairball. This is why some people don't like network analysis. You get a hairball and you say, so what? So in this particular case, we changed how we visualized the network to give us some additional insights. So this next picture, which should look like a wheel on your screen, I'm going to start trying to describe this a little bit just in case the pictures don't match. It's just another way of looking at the network with transcription factors in the middle and genes on the outside. So let me walk you through this visualization a little bit. These transcription factors in the middle are those that we had identified as having a very, very different set of edges going out in the angiogenic subtype compared to the non-angiogenic subtype. So for example, NF kappa B1 is right here. It's colored red on the outside because it had mostly red edges coming out of it. And these edges are colored based on whether or not they're specific to the angiogenic network, or the blue is the non-angiogenic network. And the outside ring are the target genes, basically whether or not they have higher average expression in the angiogenic subtype or higher average expression in the non-angiogenic subtype. And looking at this picture made us realize we really need to think about 6 different sets of genes. Genes that were targeted either uniquely by a particular edge and then could go either up in expression or down in expression related to that targeting. And then sets of genes that are targeted by both networks, both in the angiogenic and non-angiogenic. The other thing this network layout led us to realize is this cluster of edges right here. There were some genes that were highly targeted by pairs of transcription factors, which made a lot of sense in the context of the PANDA network algorithm, which we actually were trying to model co-regulation by pairs of genes, by pairs of transcription factors. That's the protein-protein interaction data that we baked in there. So based on that analysis, we did a co-targeting analysis of each of these 6 sets of genes. And for the A+ edges, which are the edges that were more highly expressed in the angiogenic network and were targeted by angiogenic specific edges, we found exactly what we expected based on this network picture, right? High co-targeting by ARID3A, SOX5, and NKX2-5. Oh, do I have the wrong one? PRRX2, there it is right here. High co-targeting by them. But then we found something really interesting when we looked at what we called the N- edges, which are right here. These are genes that are more highly expressed in the angiogenic subtype, but more highly targeted in the non-angiogenic subtype. We found some of these same key transcription factors that we found previously, but we also found co-targeting by other transcription factors that didn't seem to have as high of differences in the networks as we expected. So we used this to dig in a little bit more into the underlying biology to try to figure out what might be going on. And this led us to a very, very interesting discovery. So this is the same little picture that you saw in the previous slide. What we found was as follows. Some of these transcription factors have known behaviors. In particular, we found ARNT and ETS1 right here. And by digging the literature, we found that these two proteins, or these two transcription factors, tend to dimerize with HIF alpha proteins. ARNT dimerizes with HIF1 alpha, and ETS1 dimerizes with HIF2 alpha. And this dimerization leads to VEGF production and angiogenesis. So their involvement in mediating something related to angiogenesis made a lot of sense. We also found that that AHR was actually in competition with HIF alpha proteins. In other words, ARNT and ETS1, the binding domain that they use to dimerize with HIF alpha proteins is actually the same binding domain that they use to dimerize with AHR. So when ARNT dimerizes with AHR, that prevents HIF alpha dimerization, and when ETS1 dimerizes with AHR, that prevents HIF alpha dimerization. That led us to hypothesize that this could be used to actually develop new treatments for people with the more aggressive angiogenic subtype in ovarian cancer. So what we did is we got publicly available expression data. We were not able to test this on our own, but we were able to get a lot of really good publicly available expression data. Let me walk you through this very briefly. So this top row is basically publicly available expression data, and I believe it was a breast cancer cell line, where they had been given a sham treatment, essentially. There was no treatment, it was the control test. And we see no differential expression of these gene sets, the ones we found were important to our network, in this no treatment. These next two lines, cisplatin and oxaliplatin, are standard treatments for ovarian cancer. Interestingly, we found that the A+ genes, the ones that go up in angiogenesis and are targeted there, didn't really have an effect. But not in the right way. Some of these other genes did actually increase their expression upon treatment. Sorafenib is a known anti-angiogenesis drug. It's the type of drug someone is given that has a tumor that's already progressed to angiogenesis, independent of tumor type. And we saw that the drug, like expected, all the genes went the opposite direction. So this is our negative control, our positive control, and standard treatments. Here are our proposed treatments. Basically, we wanted to say we want to have anti-HIF, we want to prevent HIF protein from being produced, and we want to increase AHR. And we found in all the publicly available data sets that we tested that the genes went in a direction consistent with even the sorafenib treatment. And so what we were hypothesizing is that combining these two together would be even more powerful and could definitely be used in the context of treating an angiogenic subtype in ovarian cancer. Okay. So that wraps up gene regulatory networks. So how do we summarize this? What do networks teach us about disease? Well, kind of getting back to the original treatment, the whole hope of what I just showed you is that we know that originally we have what we call a normal tissue network. Our networks are different, of course, but they're all hopefully behaving fairly normally, fairly healthily. Over time, what happens is people get sick, but we get sick in different ways. And hopefully through using something like this approach, we can identify differences in the networks and use them for developing treatments. Now, Crystal mentioned something very early on in her introduction to me, and that is that even this is a fairly limited approach, right? Sure, this makes a lot of sense when we have exactly two subtypes, but we all know that diseases don't fit nicely and neatly into these little buckets, right? I made the argument for Susan and Jane, but what happens if John, Bill, and Tom walk in? Don't they have different networks? What if they are a different age? What if they have different environmental exposures? What if they smoke or don't smoke? What if one is obese versus one is a healthy weight? Will that impact the regulatory networks, and will that impact how we need to treat them? And so kind of to tackle this issue, several years ago, I developed another approach. I'll get into detail on the next slide, but it's to reverse engineer sample-specific networks or individual networks. And in order to motivate that approach, I like to kind of take a step back and think a little bit how we got our networks to begin with. Well, how do we get the network to begin with? We started with something like gene expression data, which measured information across all genes and all samples. We combined that with other regulatory information and we got one network, for example, for the angiogenic subtype. That network represents information from all the input samples, all the people who had been designated to that subtype, right? All the ones that had been designated as having a treatment response. But we know that these samples often represent a diverse set of conditions or people, right? There's many confounding factors. There's many covariates we need to consider. What we did is we developed this method, Linus, that will take an aggregate network and basically decompose that aggregate network into networks corresponding to each of the input samples. How does it do that? Well, it does that as follows. Let's say we have something like input gene expression data. Over here, the rows are the genes, the columns are the samples. What we can do is we can use that input gene expression data to develop or reconstruct a network. This network represents contributions from every single input sample. Now let's say we take the exact same gene expression data and throw one out. We can use that expression data missing one sample to reconstruct a second network, which basically represents the contributions from all samples except for Q, right? The one that we excluded. And if you make some underlying assumptions and do a long derivation, you can actually use these two aggregate networks reconstructed using something like Panda, could even be something like Pearson correlation, WGCNA, Arachne, CLR, Seren, take your choice. And we can use that to solve for the individual sample network. And we do that as follows. First, we subtract these two aggregate networks, the one representing everybody and the one representing everyone minus one. This now represents the sample Qs, or the individual samples' contribution to the overall aggregate network. But we don't just want the difference network, we want the whole network. So we have to scale that appropriately first, right? So if we build a network with 100 samples and 99, that's different than building it with 1,000 samples and 999, right? The more samples you have, the smaller the differences, and so the more you have to scale it. So we have to scale it by the number of samples so that we bring it up to the correct unit space. And then we add in the network estimated without sample Q. This is essentially the background network that's shared by everyone. This now gives you sample Q's individual network. This method was published in 2019, and both prior to publication and along the way, because it took us a while to convince reviewers that the math was right. We applied this into a number of different applications. And again, I wanted this talk to have a little bit more of a medicine focus. So I wanted to highlight one particular application where we applied the Linus method to look at sex-linked differences in colon cancer drug metabolism, basically drug response. So what did we do in this particular application? Well, this is the overall framework. This is from the paper. So again, if you're interested, you're welcome to look it up. What we did is we took input protein-protein interaction data, transcription factor to gene target information, and gene expression data, and we used Panda to infer aggregate networks. We then applied Linus to these aggregate networks to get individual sample networks, right? And what we did is we applied Panda and Linus to estimate these networks for each individual sample in 2 colon cancer data sets. The first data set was the discovery data set from TCGA. In this case, we had 207 females and 238 males. This is a nice set because it was all consistently done. It was RNA sequencing data. We had fairly consistent information from it because it was from the Cancer Genome Atlas. Now, we also wanted to see how well our results replicated in different data sets. And so what the lead author did in this particular paper, Camila Lopez Ramos, is she took a number of expression data sets from the Gene Expression Omnibus that were all done on the same microRNA data set. We wanted to stay within at least the same platform. And she was able to combine multiple Gene Expression Omnibus data sets and ultimately get 572 additional female networks for females with colon cancer, an additional 621 male networks for males that had a diagnosed case of colon cancer. So we have lots and lots of networks here, 200 plus 572 females, 238 plus 621 males. what do we do with this? Well, what we wanted to do is we wanted to compare these networks. And one thing that's very nice about Linus is that you can apply most biostatistical methods that you're used to applying to gene expression data to the networks themselves. So what Camilla did is she took every single individual sample network and she calculated within that network gene targeting scores for all the genes. The way she did that is she took all the individual sample edge weights, so the weight at the edge going into that gene in the network, and she summed them up, essentially a strength or a weighted degree. So we ended up having for every single individual a score for a gene that represented how much targeting of that gene existed in that individual sample network. She then performed a differential gene target analysis using lemma. She corrected for stage, age, and race. I mean, again, we have individual sample networks, so we can correct for these covariates that are actually really important for understanding differences. And she found genes that were differentially targeted between men and women. And one of the top ones that she found in both the TCGA data set and the GEO data set, and again, this is why it was really important to have the replication population, she found it in both, was the KEGG drug metabolism pathway. So here is an illustration of the drug metabolism pathway. The genes in the pathway are shown in the rows. The discovery data set, the TCGA data sets in the first column, and the validation data sets in the second column. And the color right here represents the differential targeting score between men and women that have colon cancer. And what you can see is that genes in the drug metabolism pathway seem to be much more strongly targeted in females compared to males, at least overall. Not every single gene is high, but it was enough to give us a significant enrichment using something like gene set enrichment analysis. Okay, so what, right? We found this thing that this makes a lot of sense. What do we do with this, right? Well, we wanted to understand if this had clinical relevance. So what Camilla did is she took all the individuals and she broke them into half, into two groups, based on whether or not they had more targeting of the drug metabolism pathway or less targeting of the drug metabolism pathway. And then she ran a survival analysis separately for males and females. So over here on the left, you see males that have low targeting of the pathway and males that have high targeting of the pathway. And we don't see a huge difference in survival. But in females, those who have high targeting of the pathway have a significantly higher overall survival than those that have low targeting of the pathway. And this makes a lot of sense, right? Females who are able to metabolize the chemotherapy drugs are able to respond to the chemotherapy drugs, and they will have overall survival. Or higher survival, which gives us a way to have a really good precision medicine application of these individual sample networks, right? If someone can walk into the clinic, we can say, this is your network, this is the treatment you need. So to wrap up, to kind of bring you back to where we all started, is this the future of network medicine, right? If Jane and Susan both walked into the clinic and they were both ill, could we actually go and say, well, we know that you started with a healthy network, but now your network is this. And based on that, we know you should be treated differently, right? Maybe, Jane, you have high targeting of this drug metabolism pathway, so you can have a standard chemotherapy drug. Maybe Susan doesn't. She needs some other line of treatment, right? And hopefully, at the end of the day, we can give them different treatments that actually target the biological processes that are active in their cells, and they both get better. So that wraps up the main part of my talk. I have 5 more minutes, which is perfect timing, because I did want to spend a few minutes talking about the next steps, right? So I started this whole thing talking about the importance of biological processes, the importance of gene regulation. And anyone who's paying really, really close attention soon realized I really only talked about 3 main mechanisms for gene regulation, right? Protein-protein interactions between pairs of transcription factors, where those transcription factors might bind in the genome, and ultimately mRNA gene expression. But I recently showed a picture that had a lot more complexity to it. And this complexity is important. We've just kind of skipped it over, right? We pretended it doesn't exist, but it's there. So I wanted to highlight 2 future directions of algorithms and methods that have recently been published that we're hoping to use more and more and more, so that when you come back and listen to me talk in 2 years, I'll have more application papers using these other methods. And that's highlighting microRNA expression and microRNA binding, as well as chromatin state. So incorporating microRNAs into PANDA using PUMA. So PUMA stands for Panda Using MicroRNA Association, so it's a nice, easy acronym. So essentially what PUMA does is it incorporates both transcription factors and microRNAs as potential regulators of downstream target genes. If you think about that middle bipartite network between regulators and genes, basically you have both TF regulators and microRNAs. And it modifies some of the message passing equation to better model the mechanisms by which microRNAs work together or don't work together, and same thing with transcription factors. We applied PUMA to study tissue specificity using the GTEx RNA-seq data, and we were able to identify microRNAs that are regulating tissue-specific processes, right? So, for example, this figure over here is a picture from the paper, and here's a good one. In testes, we were finding microRNAs that were specifically regulating pathways such as sperm motility, things that made a lot of sense. Right, in the vagina, we had ovulation cycle and rhythmic process, a lot of things that make sense. And then Marika Kujer, who led this project, was able to go through and look at the microRNAs that actually made up these individual edges and look into various databases. And we found evidence that those particular microRNAs, when mutated, actually can result in disease development in that tissue. It was a really, really nice analysis. Another method I wanted to highlight is incorporating epigenetic data using the SPIDER method. And this is a method I'm actually really excited about because I think ignoring the epigenetic data is a huge miss in the original network. I know why we started with gene expression. I know why we started with the other things. It was widely available. It's what everyone uses. But I think epigenetics plays a really fundamental role in gene regulation. We need to think about it more. So what does SPIDER do? Well, SPIDER filters the input data used by PANDA using chromatin information. Basically, if a transcription factor binding site is an area of open chromatin, we keep the edge from the transcription factor to the gene that is adjacent to that open chromatin. But if the transcription factor binding site is not an open chromatin, we prune that edge. It just changes that input information. We've applied SPIDER to DNase hypersensitivity data, which is data that measures open and closed chromatin in 6 cell lines from ENCODE. These are the 6 cell lines over here. This is A549, stem cell line, I think H1-HESC, liver cancer, blood cells, cervical cancer, and blood cancer. And what we found, and this is the SPIDER regulatory network on the far end here, is that SPIDER predicted much, much more highly accurate and cell type specific networks than the baseline networks that didn't incorporate epigenetic information, which are the ones shown on the right. And these are benchmarked using chip sequencing data. They also uncovered what we called hidden interactions, edges between transcription factors and genes where there's not a binding site for that transcription factor in the promoter region of the gene. So we were very excited about this method. It's now accepted and in press at MPJ Systems Biology. So hopefully that'll be coming out soon and you can read it. It's also on the archive or the bioarchive, I believe, for anyone who's interested. All right, so conclusions and to wrap up. Networks are a powerful framework for modeling biological systems. I hope that you leave this talk excited about networks and understanding that even though they're very complex, even though you get a hairball sometimes, they can lead us into insights and hypotheses that we wouldn't have found before. Analyzing these networks can lead to important insights into disease processes, and in particular, comparing these networks can lead to important insights into disease processes, right? Every single analysis I showed you wasn't just looking at one big hairball network. It was saying, what are the differences between these? All our cells share some things in common, but it's what makes us different that leads to different type of outcomes, and that's what's really important in precision medicine. And finally, final conclusion, it's important when we to develop regulatory network models that can effectively integrate multiple types of omics data, right? So I know I only touched on it very briefly there at the end, but especially in that epigenetic application, we can really see that the accuracy of the networks we're predicting increases when we incorporate more and more and relevant biological mechanisms. And I will just say as a note that it's challenging to effectively integrate them. We tried lots of different variations before we landed on how we did it. And so it's really important to spend some time and thought to figuring out how these mechanisms work together. So with that, I'll put up my acknowledgement slides. I clearly didn't do this work by myself. I highlighted a few people already, such as Camilla and Marika, who did a lot of work both in the lioness and the colon cancer network analysis. Other collaborators on the COPD, asthma, and ovarian cancer analysis, and Abhijit, who developed Spider, and Marika helped a lot with Puma. And with that, I will take questions. I'll put up my conclusion slide because I think that's a better slide to have up than acknowledgements for questions.
Thank you very much for this exciting talk. I do not think you need to convince anyone, at least within TranSYS, about the importance of networks. If there is one common theme or two common themes, it's precision medicine and network analysis at some point. I'm looking at the chat. Hang on. Yes, there is a question. Are there any applications of gene regulatory networks using 3D chromatin structure?
Oh, that's an excellent question. So I actually had an undergraduate student who worked with me this past summer to try to get 3D chromatin structure data embedded into Panda. I don't know of good ones right now, but I can tell you that what we were trying to do with her, and we got some really promising initial results, is basically use high-C data to map which enhancers are physically close to promoter regions, and then add in the edges from transcription factors to genes where the transcription factors are actually in the enhancer region, and that enhancer region is close to the promoter. So that is definitely a work in progress. We were kind of to leverage off of what we already started with the spider algorithm and build off of that. So yeah, I think that's really important. Yeah, so not yet. But yeah, excellent question, and definitely a future direction that I didn't even touch on.
With respect to that answer, you said we are adapting Panda in that direction. So do you mean it each time needs to be adapted if you have another data type included, or is it generic enough? Because basically you need relationships, right?
Yeah, that's a really, really good question. So it is fairly generic, and we could probably just use it as is. But when I say adapted, I mean really systematically going through and trying to understand how the data going into the model kind of propagates and causes things to change. So as an example, when we first put the DNase hypersensitivity data in the model, if you put it in there very, very naively and just prune it, which is how I explained it because that's the simple thing, you get that the overall AUC is really improved, but that you actually lose some accuracy among your highest weight edges. Your highest weight edges actually do worse, which was really, really concerning for me because that's how people want to analyze networks, is take the top high weight edges. They don't care if the overall network is accurate. So we actually had to do some tweaking in Spider about how to reweight those input edges with the DNase data in order to get something that I felt was very, very biologically meaningful. The same thing with the 3D chromatin data. One thing that I think will need to be considered, and we didn't get this far in the project, but a gene that has many, many enhancers will have many, many transcription factors targeting it because there'll be more effective DNA around it, and it might become a very high degree node in your initial network. And that might be a good thing, or it could just be saturating the information. So when I say adapt, the mathematics is very, very generalizable, but understanding how individual predictions around something like high degree nodes, things that have many enhancers, few enhancers, lots of chromatin data, etc., is tricky.
So it's basically the pre-processing before you, okay.
Yeah, it's the pre-processing.
Yeah. Okay. Yeah. There is another question actually from the same individual, Julie Segueni. Oh, wait a minute, now it's moving. Why focus on TF enhancer interaction and not enhancer promoter loops? So what's the motivation? Yeah.
Right, so this would actually have both, and I'm sorry I don't have a slide to illustrate this. So we'd be focusing on enhancer promoter loops, identifying the loops, and then we would draw the interaction from the transcription factor to the gene to actually build the regulatory network. And the reason so I guess to answer your question, we do look at enhancer promoter loops, right? And that is our main area of focus. But what we're actually aiming to construct and analyze is transcription factor to gene networks and not enhancer to promoter networks. And the reason we're focusing on that long term is because we really want to get at this idea of biological processes. What are the genes doing? Because that's what manifests as phenotype, and that's what we can kind of think about targeting in a precision medicine framework. I don't know if that answers the question, but hopefully it does.
Mm-hmm. And there was also a question about, hang on, I'm trying to read it. Oh, that's a long one. Okay, from ESR 9 for Linus, why going into the trouble of okay, it's jumping, I apologize. Why going into the trouble of recreating a network for a single individual instead of creating it from a single sample from the expression data? And yeah, and how did you compare the differences between the networks? Which algorithm? And then I'll go to the next question.
Right, I'll try to answer that.
Yeah.
So I tend to use individual person, individual sample interchangeably. And I apologize for that slippage of language. So Linus always calculates the networks for individual samples. Or it doesn't have to, but that's how we've always applied it. In our original methods paper, and if you want to, you can look it up. It's the one that's in iScience in 2019. We actually applied Linus to a data set where we had technical replicates of people. And so I forget the exact numbers, but I think it was something like 60 individuals with technical replicates, so 120 sample-specific networks. And we were able to see, is the sample-specific network for the same person that's a technical replicate at least closer than the networks for two different individuals or two different people. And so I think the reason to, and that application, basically what we did, just to kind of take you to the end, is we built the individual sample networks. And then once we confirmed that the two samples from the same individual were consistent, we averaged those together to do all our downstream analysis, right? So I think that ultimately for precision medicine, we're going to want to probably focus on individual people and networks, individual subject networks. But in data sets, just practically, we tend to do single sample networks, and then we use any sort of replicates in there as quality control. Again, I hope that answered the question.
And there is another one. So from ESR 10, I was actually trying to also follow by reading the paper during the presentation, but it's really lots of info. Do you think part of the method can be used in the context of multiplex networks? Would it be possible to contact you once all the info is digested? I guess yes. But so this question is whether it can also be used in the context of multiplex networks.
You mean the Panda method can be used in the context of multiplex networks?
I think it was in the same realm of Linus.
Oh, interesting. So I do think so. This is an area that I haven't spent as much time on. So there is a paper that I believe Marika Kruger was on, and I'm sorry because I wasn't on the paper. She's now gone and started her own lab, and she helped a lot with Linus, where they actually did apply Linus to build sample-specific networks, and then applied, I believe, a multiplex community structure approach to find shared community structure across networks. I do think that it definitely has that ability, and it's something that even I've toyed with, every 6 months when I get super excited and just want to do things on the side. I think there's a lot of computational issues that would need to be overcome in order to do that. And in particular, the networks estimated by Linus are actually complete. They're fully dense, so you would need to threshold them. And that might seem trivial, but what we found, at least in biological applications, is a lot of times the most important differences in these networks are kind of subtle and in what I would call medium weight edges. Right? The edges that are high weight across everyone tend to be cell cycle processes and cell metabolism processes, kind of boring. And then the edges that seem to be varying the most are the ones that are medium weight and tend to be more disease relevant. And so short answer, yes, I do think it could be used in the context of multiplex network. I think that there would need to be some pre-processing steps that would need to be thought about in order to use it effectively.
Yeah. of course, when you're starting to interpret the individual specific networks, then you can, then you have to treat them right as a multiplex on their own. It's not for the construction, but for the interpretation, which is also actually challenging, as we are finding out in the lab. There was another question from one of our ESRs, Marina. So she's using Linus very intensively and was wondering if you're going to make a pipeline out of it to improve the usability of the tool. It's a very promising package, she writes, but also a little bit difficult to operate with because of few technical disadvantages.
So are you using the one on NetZoo? And if so, if you're having any issues, feel free to contact me and I can put you directly in touch with the maintainer, because this is supposed to be it's as streamlined as possible. We even have a GPU version of Linus, which should make it more computationally efficient. And I'm happy to point you to the most recent implementation. And if you're not already using that, put you in contact with the main maintainer right now to make sure that it's running smoothly.
Yeah, she doesn't use NetZoo. She uses it like an independent package.
Okay, yeah, if you're using the independent package from R, the one from the Linus R paper, I would say your main issue is probably just that R is not a very efficient language for doing these things. So put me in contact. We have versions in Python and we have versions implemented in GPU, which will make things much more effective. Yeah,
and I've also seen a question. Do you think that omics data at single cell level could help to infer patient-specific networks?
Well, those are two very different questions in my mind. So could single cell data help infer patient-specific networks? So I think you can get single cell data from individual patients. And let me tell you about one application where we started doing this. Again, it's still in the early stages. So we were able to basically take single cell data that had been collected on patients with asthma or control. So we had asthma and control subjects and single cell data in each of these. I think it was like 15 to 20 total subjects, not huge, but a lot for single cell data. And what we did is we basically employed a sparsity aware version of Linus to estimate cell type specific correlation networks in each individual person. So we had a cell type and person specific correlation network, quote unquote. We didn't wrap it around Panda, we wrapped it around the correlation. And then what we had started doing is we started taking a cell type specific network in an individual and comparing between asthma subjects and controls to see if there are certain cell types networks that were different between the different groups. Unfortunately, the postdoc who was working on that project left, and I'm waiting on someone to pick it up. So the short answer to your question is yes, it could definitely be useful, and it's something that we've started doing, but I don't have anything that is final and published to point you to yet.
Yeah.
But if you're interested, contact me.
And a related question, I think, is when we are using individual specific networks, of course, you need to think about interpretation, right? And it's always very hard, I feel, but maybe it's me, if you want to explain this to other people, what exactly, it is, it can entail. But so if I talk to pharma, for instance, then they say, oh, yeah, validation, etc. Well, you have to go to the single cells and you have to see, what you see in there. But of course, these are two different things, right? The bulk and the single cell. But what is your take on that? To what extent could the results on single cells actually help us perhaps to validate, the individual specific networks constructed from bulk data?
Well, I think that the single cell data could give us some insights into what cell types need to be used for the validation. If we have individual sample networks that are generated in bulk, which is what the majority of patient data cohorts are in, and we find something that's really promising, the question is always, okay, now we need to actually test it in the lab. And to test in the lab, we need to order some sort of cell line, and we need to know what type of cell it is. And so one thing I've talked about doing with my collaborators at the Channing is when we find something that is really different between, say, COPD cases and controls, to then look at the genes that are more targeted and see if those genes are more specific to a particular cell type that's important for something like COPD, and then use that cell type for the validation. And that's where single cell data can actually be really helpful, even if you only have it on a handful of subjects, because then you can say, okay, here are the genes we think are important in the networks and the individual networks. And then here's our single cell data for some of these subjects. And are these genes at least expressed in our cell type of interest where we're going to do the validation?
Yep.
So we haven't touched it yet, but that's how we've been thinking about it.
Right, so I see also a question, how can we interpret the individual edges having extreme high or low values?
Yeah, I think edge weight interpretation is one of the trickiest things about not only these methods, but almost any network inference method.
Yeah.
So the way I tend to explain this, at least to my biological colleagues, is the higher edge weights give us more confidence, but that doesn't mean that the lower or medium level edge weights are not confident. I also tried to explain it a little bit more in terms of something like gene expression data, because it's something that people often have a handle on. So if you think about it, a gene that has a gene expression level of 15 across everyone, well, we know it's really, really expressed. But if another gene has an expression of 8 to 9 across everyone, it still might be really important that gene is really differently expressed, right? 7 in a bunch of people and 10 in a bunch of other people. But we know that a gene that wiggles around 0 to 1, it's probably either not expressed or there's something technical that's preventing us from capturing it. So I think the way to interpret edge weights is actually kind of similar, right? We know the edge is really, really likely to be there if it's a really, really high edge weight. That doesn't necessarily mean it's the most critical, the most important for the actual biological process. And that's why I really emphasize comparing networks a lot, because I do think that these edge weights are kind of squishy to what they actually mean. And that the most relevant ones are the ones that come up as statistically different between different patient groups.
Maybe a few last questions. So it's again about ISN. There was a question about, can this be curtailed or used in the context of directed networks? So directed individual specific networks. Or if I can add on a second part of that question to it from myself, what's basically the difference between, an ISN as constructed in the Lioness framework and perhaps a personalized network where you actually have causality directions induced by, for instance, individual mutations, right? Making that network also individual specific.
Oh yeah, okay, those are two very questions. I'll try to be as brief as possible. So I think getting back to like mathematics and generalizability, Linus is supposed to be a wrapper, right? So if you have some sort of network method that generates a directed network using a set of samples, then Linus theoretically can be used. It's just an equation, right? You can throw out a sample and not throw out a sample and apply the equation, and then the network that comes out will be directed. And a good example of that is Panda. Panda actually is a directed network. It's a bipartite network, right? But it's directed. It goes from transcription factors to genes. So it definitely can be used in the context of directed network as long as the underlying aggregate reconstruction algorithm is directed and takes into account a bunch of samples. Now getting to your second question, which I think is a little bit more subtle. What about the idea of genetic mutations and building something directly from the individual data? We have a couple various projects that are investigating that directly. For example, there's another zoo animal. I'm not driving this project. I'm middle author on it, but they're using Panda, where one of my colleagues, John Plattig, also at the Channing, has basically taken the Panda framework and modified edges in the original TF to gene prior based on the existence of a genetic mutation inside a transcription factor binding site, combined with evidence of an effect on gene expression, EQTL evidence. Does that individual SNP in the TF binding site actually influence the expression of the gene? And doing this, he's able to infer genetic profile specific networks, which are individual specific networks. And so one thing he and I have talked about is ultimately figuring out how can we compare or combine Linus with this framework. So that's one project, and that's the EGRIT method. If anyone wants to Google it, the paper is under its second round of very, very minor revisions at Genome Research, so we're hoping it'll be accepted soon. The other thing I've been doing a little bit more recently is I've been taking something like a Linus inferred network, and I've been looking at the association of the edge weight with genetic profiles, right? You're basically running an edge QTL analysis. And that lets us identify subnetworks that seem to be associated with individual SNPs. So that's not a direct comparison of something like an individual specific, like genetically driven network versus a Linus network, but it is another way to combine genetic information with a Linus network to see, is there something maybe in the underlying genetics that was driving some difference in that network that we potentially should model. Yeah.
And if the epistasis field is moving a little bit further and is able to generate robust protocols, actually that would also perfectly fit in, right? Because you have connections between genetic markers, you have your gene expression, you have eQTL information, and you can, you know, use the Panda framework. Yeah. Okay, I hope I haven't missed any other question. It was sometimes difficult to follow because the chat was just hopping further, but I think I covered it all. If not, I apologize already, and we have made available a little online form where people can still enter their questions, and we will then forward them to you. That's
excellent. And I also just put up my slide with my email address right here and my website in case people want to screenshot or write that down. You're always welcome to contact me directly.
Yeah, could you send us maybe your slides, because I, for instance, still see the conclusions.
Oh my goodness, I don't know why this isn't working.
It's a delay, I think, because sometimes it appears, but only later. Yeah.
I apologize.
Yeah. No, not a problem. Okay, so thank you very much, Kimberly, and hopefully we will meet in person again soon, but we will surely contact you for other matters as well. Yeah.
Excellent. All right, thanks again for the invitation.
Thank you for joining. Yeah.